Okay, hello everyone. Welcome back. It's the afternoon sessions. Okay, so we're going to report back from the breakout session. So let me introduce myself again. I'm Jin Yang from University of Sydney in Australia, and
I'm Jong-Eun from KAIST, South Korea.
Okay, well, I'm not sure that this is a slide.
Well, we can try to report on... No, I don't think it's possible.
However, what can I make out of that? So our session, just before they're figuring out the slide, we're looking at spatial and cancer. So in fact, this is part 2 of the session yesterday. Elle did a really wonderful session on spatial biology, looking at cancer. And today, we are sort of following on that discussion. So talking about, possible flagship project that HCA Asia will be looking at. I'm going to run out of things to say if they don't get that.
It's almost done, so a few more seconds.
Okay,
yeah, it's on.
Spatial cancer session. Okay, over to you.
Up to me, okay.
Okay, I can press the slide. There we go.
Okay, so we had been continuing discussion about how to build the HCA spatial consortia. So if you hadn't done this survey yet, I'm telling this like third time today. So please go check out the QR code.
I think it's like the 10th time.
Yeah,
already.
okay, so next slide. And then we talked about what we can do for building a flagship project about the HCA Asia cancer. And in last year, 2023, Ungyang, Angkul, Yutaka, and also Natini has discussed about this conceptual project where we can work on lung and liver and colon cancer. And they actually have started some of the work collaborating, but without core funding. They are doing private funding, and it's like a bottom-up project that they are doing. And we talked about what we can do by joining them or by creating a new project.
Yeah, so the discussion is actually quite broad. Basically, we talked about how we can formulate the project and so on. And so the group actually discussed, what's the important criteria of the flagship project, the data collection, we talk about how we should collaborate, strategy, and know-how. And for inclusivity, we actually ran a mentee during the session. And so I'm just going to report back what we actually got from that session. So the cohort in this is that distribution. So we see there's a few technologists, a few biologists, but actually that session turns out to be dominated by computational people. So interesting, but just to keep in mind, sort of the discussion will also have that sort of flavored, that was the participant in our session. So the first thing about is we talk about flagship project, what's the criteria? And we remind the people it's very important that if you are interested in what there was discussed in 2023, so, they should really join 2023 project, and that was the information there. But we also was interested from the audience, how do they actually rank in terms of what they think it was important? And here is, the results from the audience, right? So there was a lot of interest in trying to look at gene and the immediate environment, and also the Asian cancer that's unique with epidemiology seems to be what people are interested in. So we discussed whether people go broad, every single cancer, or we go narrow and find something really specific to Australasia, Asia, and Australia. And so then it prompted us to ask, what's the cancer that, and that question was asked yesterday, and people during the discussion session actually speak out and actually talk about why is it that certain cancer is more unique to Asia. And anyway, that is sort of the word cloud that represent the interest in the group. And so maybe the people who are doing data generation could actually think about, what's the interest that, and I think putting this together with the survey that Xiao is actually put together will give us a scene of what is it that we are interested in. Okay, so then we go on to, if this is what we are going to go for as a flagship project, what are some of the ideas that we can work on? How do we actually collaborate? How do we share the know-how? And there was a lot of great ideas that actually go on. So some of the suggestion is creating repository, having standardization, how do we chat? How do we actually have a community platform to actually bring everyone together? How do we actually go for funding? How do we actually, implement so that, if we want to collaborate, this is more the implementation plan, right? And so there is a lot of things that's discussed, but these are also some of the things that people talk about offline. And what's most interesting was this hackathon and dream challenge that sort of came up to say, if we want to raise our profile, should we put something together, put it out as a competition? And this is coming from the fact that there's 33 computational people in the room, that maybe we could raise our profile in having some collective data out there, and that could also bridge the gap between computation, data generator and data consumer. Okay, maybe you could finish off.
Yes, I think. So did we cover the funding part? Or?
We didn't.
Yeah, okay.
But you can.
So the concluding remark on this session is about, people were actually wondering how to start spawn a flagship project like IDA. And then the basic idea was that anyone can start, anyone can find the interest, the people who are sharing the interest. But then for the authorships and everything, we should have some angle, like specific angle, for example, interesting epidemiology. For example, there was a mention about Singapore can be a very interesting site because many different genetic background can migrate in and then share the same environmental factors. So those kind of study design can really be a very unique feature about HCA Asia flagship project. And there was a lot of demand on data collection and harmonization. So I think there can be also purely computational sort of collaborative work. We're thinking about what kind of metadata that we collect and how to make the really good pipeline. And there was also questions about, will there be any HCA-based support on computing facilities, for example, because spatial data requires a lot of cloud storage and everything. So with that, I think that's the kind of conclude our session.
Yep.
Yeah.
That's the cancer and spatial session. And thank you for all the people who participate in it. That was all your ideas.
And then, yeah, thank you. And then I pass over to Yuki and Pompan for reporting infectious disease and autoimmune.
Okay, thank you for everyone coming back to the session again. So this is Yukinori Okada from the University of Tokyo and Riken from Japan, and
Pornpan Matangkasombat from Mahidol University, Thailand.
So we do have a break session for infection and autoimmune diseases. Again, infection disease has been a strong burden for us, public has everything for last plus several years. But now COVID-19 almost passed, but we know that this should become the next pandemic in not far future, and also vaccination is also still important. And immune disease and infectious disease vaccination, these are strongly correlated, sometimes positive or negative correlation. So infection and autoimmune disease are very important. So we have some discussions, and yes, again, how to see, oh, here's the rest, okay. We show some, several very many feedback from the audience, and okay, let me show the strengths. Oh, okay, we have lots of open questions. And I think there's so many cancers in blood, but I think there's so many infectious diseases and maybe much further number of infectious diseases coming. So this is of local importance: dengue, NTM, tuberculosis, autoimmune disease, and how's the comorbidity? Sometimes positive comorbidity and negative comorbidity. And of course, what disease is important, it always changes, right? WHO priority list and HCA survey. And anyway, again, infectious disease itself, autoimmune disease serve strong importance in the public health. We should definitely put it on. And also, these diseases, because we have some common interest, is because everyone knows infectious disease, right? Everyone knows COVID with others. And it means that there's so many available samples and metadata. So actually, this is or difficult to find people who haven't get COVID-19 now, right? So it is one of the strengths. We are very easy to collect so many samples. And yes, we already exist in very infrastructure genomics in immunology. For example, in terms of single-cell sequencing, it's very good to the immune profile, and also germline genetics, HCA, something raised. And yes, strong interest with community. So, and Papa, this is a, oops.
Oh, do you want to?
Oh yeah, can anyway. But also we found that through discussion we have some difficulty. So there is notable in some countries, especially infectious disease burden in many countries across the world, and some countries with non-good economical condition likely to be susceptible to infections, but they have some difficulty in the technology. So high cost in adequate funding, or not only vet things, but also machines, data analysis capability, sometimes computational, but or some biometric scale is still have inequity. And yes, the point is different diseases in different countries, but again, because of the climate changes, now the infectious diseases are expanding, right? For example, even though dengue, we recently found some patients in Japan, right? Because of the climate. Anyway, they are expanding, but still different infectious disease, different countries. We should need to make a strategy where to prioritize. Okay.
So we discussed how could we work together as a community. So because a lot of limitation and challenges that we are facing is on funding and financial support, not so much of the sample collection. I think we have great open question in infectious disease and autoimmune disease, and we talked to several researchers of several countries. We have good cohorts, we have samples collected, cryopreserved, so that's not so much of the problem. The problem is we don't have enough funding to perform the actual experiment. So we discussed how could we go about and work together in order to overcome all those unmet needs. So one issue is that we could co-apply for funding. We might leverage ourselves and have higher chance in getting funding if we collaborate from different countries in Asia or even from different regions, including like Africa and South America that might face the similar type of tropical infectious disease. That's just one example. So we could potentially apply for international funding and leverage disease burden because of the global geographic expansion that Yuki just mentioned. For example, the dengue is transmitted by mosquito vector. So with global warming, mosquito can live in like expanded geographic areas. So people in Southern Europe or Southern America also started to get it. So maybe this is an opportunity to get more funding. So, and then there's also discussion on banking sample, even though we don't have the funding yet, but it's good to start talking about potential project and start banking sample. We can share protocol and metadata that we need to collect in order to be ready when the fund comes. And then we can take some advantage of certain programs such as the Scale Bioscience program that can help resource limited setting. So in order to apply for funding, we also discuss about how it impact, how the single cell studies will have any impact, so the funder would recognize. So we talk about a biomarker, although it might not be cost effective to do single cell for biomarkers, but it could identify certain gene sets that we can potentially use as biomarkers, target discovery, vaccine design across population, global population, and immunotherapeutic. So also besides funding, there's also unmet need on certain capacity on data generation and data analysis. So working together on capacity building for local team members would be also important. We were discussing whether we could have regional expertise hub, and people from that area can like come together and use that expertise hub, and we can help each other in training and workshops such as one that HCA organized that has workshop in Latin America, Africa, and Asia. We could have interdisciplinary collaboration like human genomes and single-cell RNA-seq and immunology, bioinformatics, also vector and entomology and pathogens in order to understand like host-vector pathogen interaction. We also discussed about autoimmune disease as well as infectious disease. Autoimmune disease is also a burden within our region. And Yuki also mentioned like human genetics involvement of this autoimmune disease. So, and lastly, we could establish a platform or tools or protocol that can be shared among members so that we together can move on as a community. I think.
Yeah, good. Thank you for Pampa. Again, so these are the feedback from our break session. But we are now further expanding, recruiting many collaborators. For example, Pampa leading dengue analysis, and I'm doing NTM analysis. But if people who have any interest, let us know. And also, would like to emphasize finally is networking and community is very important in these fields. So when we have COVID-19 pandemic, we somehow have difficulty to finding colleagues. But now, under the umbrella of HCA and HCA Asia, and next pandemic, we will quickly make the team and go to research. Okay, thank you for again everyone for joining the session, and thank you for Pampa making a slide, right? It's the best slide for breakout session. Thank you.
Next, let me introduce the next session. Breakout session is from Xiao and Joseph Powell on single-cell QTL.
All right, this is the slide you saw earlier. You got a sneak peek. So for those of you who don't know me, my name is Joseph Powell. I'm a PI at the Garvan Institute and the University of New South Wales in Sydney, and reporting back on the single-cell QTL breakout along with
Shyam Prabhakar, Genome Institute of Singapore. I just a word in advance. You saw the best slides. You heard a lot about best breakout session yesterday. You ain't seen nothing yet.
It's quite a throwdown, isn't it? Okay, so we started off, I guess, having recognizing that there was a fair bit of diversity amongst the participants. A lot of people have been working on genetics, or the intersection of genetics and cellular genomics. But we covered, I guess, a series of topics. First of all, of I guess the key considerations that are important in the types of data generation analytical methods that are important in this field. So particularly the need to generate cohorts, large cohorts of single-cell data with matched genomic data to provide the relevant statistical power and, of course, representations of genetic allelic diversity within populations to start finding these kind of effects. And associated with that, the necessity of having scaled technology, particularly for generating single-cell data and the sort of approaches that you can apply to generate that. And then we spent a little bit of time, and I guess it's always woven through the breakout session in general, in that this is a very rapidly evolving area of methodological advance. And so there is particular interest within the community, and I think quite relevant to HCA, around methods that can be used to both identify QTL effects, but also how we think about classifying cell types, but probably more importantly, cell states, and how cell states would be derived based on particular biological axes, and so on and so forth. And when I give a presentation later, you'll probably see these two slides in my talk, but the point being is that this is an area where there's a generally quite rapidly evolving series of publications are coming out. You've obviously heard about different cohorts and consortiums that are occurring both within Asia and Asia-Pac, and of course more broadly across the rest of the world. So that was the kind of context and background that we discussed. And then the areas that we sort of dug into where we thought there was particular opportunities for this community to come together, and we'll come to some of the takeaway messages at the end, was thinking about using spatial approaches for doing spatial genetics. And this was particularly motivated by some of our views within the room that One, that we've generated a lot of single-cell cohorts on circulating or say, liquid samples, largely because they're accessible and relatively highly scalable. So a lot of data on PBMCs, cell line systems, and so on and so forth. But a huge gap in understanding how genetic variation impacts cell phenotypes and cell diversity in situ contexts or in solid tissues. And so we started having some early conversations about what we could do as a community to think about the data that's been generated for various spatial projects and atlasing projects, and how we could think about the methodological approaches for analyzing that. And then I think a really key thing that came out of the discussion, and I personally think this is really relevant to the local community here in Asia and Asia Pacific region, is that we've generated a number of cohorts across different geographical regions or different genetic ancestral diversity groups, but what we can do to integrate some of these initiatives and efforts to evaluate how genetic and non-genetic factors influence cell phenotypes, and some of the challenges of where you'd need to do that to split between true biological effects and just technical variation of generating data in different spaces. And then finally, we spent a fair bit of time talking about these dynamic effects. So thinking around cells, less as cell types and more as states, and then evaluating how these allelic effects vary as cells transition across these states. It's probably worth flagging a few opportunities to collaborate for those of you interested. So HCA Asia members, particularly working on outside of the tissue systems that I just mentioned, work in different liver phenotypes. There's disease focuses, obviously, in autoimmune disease. There was a really good call out, I think, from the infectious disease community in the last slides, and the cancer as well. And you could read the other subgroups there that these might be opportunities for people that weren't in the session that would be interested in collaborating with some folks on. And then the action items, I guess, that came out of this, and then I'll see if Xiao has any final comments to add to this, was that there's a really exciting, well, maybe exciting is not the right word, but important opportunity to think about standardizing protocols for metadata collection across this community, and opportunities to present some of those ideas around perspectives articles. There's obviously the requirements of how you would computationally analyze these differences between cohorts, and some potentially interesting initiatives that we could do within our local community. There was a very practical suggestion that because this is a very rapidly evolving field of technological advance and people generating more and more data, that having regular meetings or an open regular meeting within the community to share, I guess, opportunities to collaborate, new advances methodologically, new tools that are coming out, so on and so forth, would be really valuable. And there's a fantastic volunteer that's going to stand up and start organizing that, sitting in the back of this room. Hello. And then finally, and this is really, I think, a super ambitious but really interesting long-term activity of what we can do to start building spatial genetics into some of the resources that have been generated in this community. And obviously we'd need to be pretty targeted about where we'd start. So that would involve thinking about cohorts that are being generated or coming to be generated. But of course, most importantly, thinking about the methodological approaches to analyzing that. And there was obviously particular interest in what can we learn from genetics in circulating immune cells, and what is happening as those cells transition into infiltrating immune cells. And that's probably a very logical intersection with the work that's happened in circulating immune cell genetics. And with that, Xiao, have I missed anything that we need to cover? She's just been standing here looking pretty, so I've been doing all the talking.
That's my job.
Yeah.
No, that's a great summary, Joseph. I just want to emphasize a couple of points. So in Australasia, we have actually a remarkable strengths in this field. So Joseph published the first massive scale, 1,000 people, single cell QTL study in the world. Yuki's team has done amazing single cell genetics. There's Hyunjae is coming up next from Seoul National University, who's doing a lot of single cell genetics. I've named just 3 names. There's many more fantastic groups in Asia and Australia doing this kind of work.
Like Ada.
Either too, thank you. And so this is an opportunity. Single cell genetics and spatial genetics are really the future. There's only going to expand. And so we have to ask ourselves, okay, what can we do besides getting bigger and bigger cohorts to catalog more and more QTLs? So the next layer is comparisons. So compare this population to that population, this ethnicity to that ethnicity, this geography to that geography. As Joseph mentioned, circulating immune cells to spatially profiled tissue resident or tissue infiltrating immune cells and so on. So those comparisons will be the next level, and doing that at the single cell resolution. And then spatial will be transformative. So I think we have the strength in this consortium or in this network to really make a difference, and hope we can come together, maybe leapfrog straight to let's nucleate some spatial genetics consortium in HCA Asia, as let's nucleate something on comparing eQTL effect sizes across different population groups. And of course, there's an opportunity to lead in terms of writing perspective and where the field can go and how the field can work and so on. Yeah, let's work together on this.
Thank you very much.
Our final two breakout session leads who will take us to even greater heights are Jae Shin from GIS and Hyun-Jae Kim from Seoul National University, talking about skin and microbiome.
Hello again, I'm Jae Shin from GIS.
My name is Hyun-Jae Kim from Seoul. Okay, this is my third time participating in the HCA, supported by myself. I really love this community because it's open to everybody. So I just want to share what we have discussed in terms of constructing the skin atlas. So the discussion point we have was 3 points: why, how, and what. So already in the South Korean communities, we have constructed a number of inflammatory skin disease atlas. Actually, why we have to construct skin atlas, I think that the skin is one of the largest organ, and it's relatively easy to recruit the patient and samples. And at a clinical point of view, and because to collect the patient samples, we are using 2, 3, 4, 5 millimeters punch biopsy. Actually means that from the biopsy time point, we can collect the samples in a very standardized way. And the second, why Asian? So is a simple reason, because to increase the ethnic diversity. So if you think about the immunopathogenesis of a certain disease, this kind of explanation was from the kind of European, American, or kind of not Asian sample work. So we wanted to kind of make our own explanations why this disease is happening from our kind of Asian samples. Third, I think that we can find out the shared explanations that can be applied to a lot of diseases. So as a clinician point of view, let's think about atopic dermatitis. Atopic dermatitis is just atopic means the aberrant immune response to the nominal antigens. That's immunologic term. So it means that's just a strange. And dermatitis means that the skin inflammation itself. They don't give us any informations. But as you know, that after Regeneron developed alpha receptor blockade named dupilumab, strategically that drugs was applied to the atopic dermatitis because it is relatively easy to get approval with little money. But after that, those drugs are spreading to the indications, including the allergic rhinitis and allergic asthma, and so on. It means that all of that diseases were kind of the IL-4 receptor-mediated disease. So I feel like that even though the clinicians have defined the disease, but it's now time we have to reclassify or redefine the disease itself. So I think that in terms of the therapeutics point of view, I think the atopic dermatitis, allergic rhinitis, and the asthma is kind of the IL-4-mediated disease. So with these kind of effort, I think that we can reclassify the disease following the biologic informations. So next point is how. I think the most important point is to recruit clinicians in the field. Clinicians, because to get the samples, we need to at least have the cooperations from the clinicians, and they have a responsibility to the patient. So we have to explain better to the clinicians why we are doing this work. So while we are doing that, we have to make labeled data, very well controlled, well standardized, labeled clinical metadata is one of the most important things, we think. So I think still we don't have any kind of standardized clinical metadata collecting form. We do not ask the patient, the clinicians to collect the heavy kind of the informations because in the clinical point of view, that is extra work. So we have to have a consensus that what kind of informations from the clinical point of view, from the patient point of view, we have to collect. It means that I think that let's think about the EMR, electric medical record. Huge information is there, and but according to the nations, according to the hospital, we do not have a standardized kind of EMR system. So retrieving information from the different EMR system is also another effort we have to discuss. So at least I think that we need to have the standardized clinical metadata collecting from, again, I think that we agreed on it. And then the skin terms, clinical photo is important because the dermatologists are kind of defining skin, defining the disease following their naked eye. So, but those kind of informations can be converted from the clinical photos. So clinical photo we have to collect, I think. Then next, I think that everybody are interested in more the spatial data, how we can generate the spatial data. So as we discussed yesterday or the day before yesterday, and the image-based spatial and the sequencing-based spatial, both are needed. And but money, how we can save our money from while collecting the data. So what we can suggest is that let's make a very standardized FFP, the TMA, tumor microarray. So what I can suggest is that let's make a kind of 7 millimeters, 50 millimeters of the TMA, including the 15 different patient samples, and let's profile it with different commercial platforms, including like NanoString platform, 10x platform, and then special protein platform, the Maxima like that. So because the company are just making the machines with their conveniences, so we have to somewhat to that. And then the next part is that with those data, who will be and how we can make the standardized data analytic pipelines. So I think that a lot of good scientists have to do it together, I think. So those platforms should be open and everybody can use it. And then after that, I think that with those effort were combined, I think we can do like 3 kind of points. So the target, let's think about the target in drug development side. So we human beings have already know the good targets for the drug development, but definitely with those effort, we can kind of identify a noble targets. It means that we can accelerate the drug development process, and that can help the patient. And second point is that the patient stratification. As is patient stratification means diagnosis. If we are stratifying the patient with as is data, that means the diagnosis. If we are stratifying patient in the future, it means they need treatment. So with those efforts, I think that we can develop the biomarker-driven diagnosis, and that we can predict the patient future with this effort. So we are all open, and we discussed this point in the Korean Society yesterday, and we all agreed with it. So please, let's do it together.
maybe I'll just, mindful of time. Thank you, Hyun-jae. Obviously, he's a clinician, dermatology background, and immunology as well. I think he highlighted the importance of including clinicians and building a disease atlas. But also, why skin is that I've had opportunity to speak with Hyun-jae, who had amount of access to clinical FFPE samples. We have started a whole skin project in Singapore to study skin aging, and there are access to hundreds of samples. In Japan, we also have access to large number from Keio University. And China, we just met a member from China who has access to also large sample collection. So it's already there, and there's a huge amount of opportunity to initiate this project right away. And that once we build this, you just heard from Sham and Joseph of spatial QTL. This is probably one of the most solid tissue consideration for spatial QTL as well. So I think opportunity is tremendous, and hopefully we can work with you and be able to build this atlas together. Okay, all right, thank you.
Good afternoon, everyone. Can you hear me? So welcome to the session on human development, aging, and regeneration. So my name is Mu He from the University of Hong Kong. I'm one of the organizers for the HCA Asia in Hong Kong this year, and I'm co-host the session with Jonathan Loh from A-STAR. We heard a lot about infectious disease and cancers, and for this session, we're gonna use single-cell approaches to understand development to regeneration, which are important for all of us. So our first speaker is Zhenyu Xiao from the Beijing Institute of Biotechnology, China. She's an early career scientist, and she's gonna tell us about human early development.
Okay, thank you for the introduction, and today is my great pleasure to be here to share our recent work with you. And as the host said, I just started my own lab in Beijing Institute of Technology since 2002. And today I'm going to tell you about how I use the power of single-cell omics to decipher the secret underlying the human placenta and embryonic development. So as we all know that having a baby should be one of the most fulfilling moments in life, but this is not always the case. As we all know that a very healthy young couple can only have 40% of chance to having their baby per month, and the pathological conditions, the infertility rate is still very high, almost 20%. And a lot of these pregnancy loss happened during or pre-implantation stage. And even you are very lucky to got pregnant, you still have a big chance of carrying a baby with birth defect. And also for the pregnant mama, the life is not that easier. They have a big chance to suffer from a lot of these pregnancy-related complications. So we could partially link those pregnant-related complications to a very unknown but very important organs, which is the human placenta. And we all know that the placenta and the embryos, they all come from the fertilized egg, which is the zygote, but their lifespan are quite different. For the placenta, they can only survive for 10 months during pregnant. However, though it is this 10 months journey that can not only support your health during pregnancy, but also guarantees the health life of the mother and the baby after birth. So sitting in the maternal-fetal interface, the placenta venous is the functional unit of the human placenta. There are a lot of cell types within this interface. The major two types are the trophoblast cell lineage, which is the cytotrophoblast cells, we consider it as the stem cell-like cells that can differentiate into the extravenous trophoblast cells, and also two very amazing syncytial, which is a multinuclear cell types, the STB, the syncytial trophoblast, and also another mysterious cell type, placenta bed giant cells. And since I started my own lab, I'm fascinated by the venous core cells containing endothelial cells. The only immune cells within the human placenta, which is the Hofbauer, is a macrophage. It's a tissue resident macrophage. So the development journey of the placenta starts with the implantation of the blastocyst. And as we all know that the trophoblast lineage comes from the trophectoderm of the blastocyst. However, it is still under huge debate that where does those venous core cells come from? People said that it might come from the hypoblast lineage pre-gastrulation, or maybe it comes from the gastrulating epiblast cells after gastrulation. So much thanks to the single-cell omics, we can now join a very nice picture by our own, just like George Thierry. So with the power of the single-cell omics, I started my journey with this huge human syncytial cells, which is the STB, because according to research, it contains 58 billion nuclei within one single cytoplasm at term. And also still we don't know the differentiation trajectory of the STB nuclei. We hypothesize that all the matured and functional STB nuclei comes from the very naive fusion-competent CTB cells, but we still don't know whether it's true or not. So I started my journey with the placenta using the single-cell RNA-seq. And to my surprise, or sadly, as you can see here, with this whole human placenta atlas, we only got a very tiny bit of STB nuclei in the atlas, which is not far from enough for us to really investigate the STB features and the functions. However, although the STB journey is not that good, we're still using this map and find a very interesting, we identified the origination of the placenta Hofbauer cells, and my very talented new PhD, Jong-Eun is here with his poster outside. If you are interested in the venous core cells, its origination and the functions, please go to his poster. And I will continue my journey with STB. Thanks to my colleagues, we changed our strategy to the single-nuclear RNA and the single-nuclear ataxic, because we realized that with this multi-syncytial, we cannot use the single-cell RNA-seq. And we got a very satisfactory results. As you can see here, we got a large amount of STB nuclei and with gestation-specific dynamics. As you can see here, at early pregnancy, all those STB nuclei, their function linked to the vasculation, response to hypoxia, which makes sense because we know that during early pregnancy, all the babies and the placenta, they are under a hypoxia environment, and those hypoxia environment is good for the development of the vascular system for the baby. And at term, the STB nuclear function goes into the hormone secretion and the labors, which also makes sense. And with the power of the single-nuclear ataxic, we identified a huge amount of lower master regulators that can push the development of the STB nuclei along pregnancy. We then focused our study on the early pregnancy, of course, because a lot of birth defect and also pregnancy loss happened at this stage. So to our surprise, we identified at least 3 groups of STB nuclei at early pregnancy with specific gene expressions like SS3-TC2, PAPPA, and FLT1. And our immunostaining confirmed the existence of these 3 big groups of STB nuclear subtypes. So with these results, we questioned whether those STB nuclei follow this one single differentiation trajectory as mentioned by other research. The answer is no. By a lot of like molecular 3 RNA velocity, we used several analytical methods to investigate the development trajectory of the STB nuclei. And to our surprise, at least at early pregnancy, those STB nuclear development follows a bifurcated development trajectory with different functions. Like one group of STB nuclei, they are quite worked in the vascular development and also response to the hypoxia. And another big group of these STB nuclei that function in the hormone secretion. Of course, thanks to the integration of the single-nuclear RNA-seq and then single-nuclear attack-seq, we mapped a lot of accessibility regions along this amazing bifurcated trajectory. And we identified, of course, several specific or unique transcription factors that responsible for the development of the STB nuclei at earlier stage. Also, we drew a very nice gene regulatory network, and with a focus on several transcriptional factors like state 5A, like FOSL2, and with their enhancer and downstream genes. And also the immunostaining confirmed the coexistence of the transcriptional factor with its regulated genes. So in the lab, we now, using this data set, we constructed our own trophoblast stem cells, and also we constructed those trophoblast stem cells-based placenta organoid in the lab. So with this mini placenta in the lab, we can now do a lot of interesting work to test some medicines and also do the gene manipulation to monitor all those pregnancy-related diseases. And also, we are quite curious about to what extent our mini placenta in the lab can mimic those in vivo STB nucleus. To our surprise, of course, the mini placenta in the lab can partially mimic the marked gene expressions, as you can see in the heat map. They're quite similar to the in vivo ones. However, when we differentiated our mini placenta in the lab, we can not monitor those most matured and functional STB nuclei in the placenta organoid. So since we got those gene regulatory network, we mapped those master regulators. So we are now gene manipulating all those master regulators, and we can finally push the maturation and the function of the mini placenta in the tube. So more interestingly, at late pregnancy, those STB nuclei, the trajectory that's responsible for the vasculation and also for the hypoxia responses, they disappear. So all the STB nuclei follow this one single differentiation trajectory that's from a very naive state to a very mature state. And we are now working very hard to try to monitor this single trajectory in the lab. And with this single-cell RNA-seq and single-nuclear attack-seq techniques, we are now drawing this very nice map of the STB nuclear map in the lab. And I think, it's quite good that those in vivo datasets can actually guide the construction of the in vitro models. So now, when I started my own lab, I focused on another very interesting part of the human embryonic development, which is the third week of the human embryo development. So together with, encouraged by my postdoc supervisor, Professor Wang Hongmei in CAS, and also my PhD supervisor in Leiden University Medical Center, Susanna, we spent 4 years, and we finally got all those precious human tiny embryos in the third week of pregnancy. And now we can draw very nice cell atlas, not only for the placenta, but also for the embryos, and another very mysterious extra embryonic organs, which is the yolk sac. And because we used a lot of embryos, placenta, and of course the yolk sac and the strict ethical constraints, and of course we got a huge data set. So my student tell me that all those data, our computer cannot survive all those data sets. So we decided to investigate those events one embryo by one embryo. And my first curious focused on the axial formation of the third week of human embryo development, and also with very ancient anatomic structures, which is the notochord. So notochord sits in the middle of the human embryos, and it secretes the sonic Hae-Ock signals to pattern the anterior, posterior, left, right, and dorsal ventral axis of the human embryos. So we started our story with the very precious human Carnegie stage 8 human embryos, which is an late gastrulars during early human embryonic development. We cherished these embryos very much, so we decided to use the most expensive way to really investigate these embryos. So we sectioned those embryos along the transverse plane, and we finally got 62 serious transverse plane sections. We put those on the sequence chips, and together with all the data sets, we used the Maya strategy to construct those human Carnegie stage 8 embryos in the lab. And of course, to our surprise, because in 2021, Tizer published their work on a very early human gastrulars, which is a Carnegie stage 7 human embryos. They tell us a lot of lineage-specific events. However, in their data sets, because of the loss of the spatial information, they can only define those mesoderm cells by the time, because as you can see here, they use the nascent, emergent, and the advanced to define mesoderm cells. And to our knowledge, mesoderm should be defined by their location, right? So we got a very nice notochord sitting in the middle of the human embryos, and we finally defined all those mesoderm cells patterning along the notochord, and we can name them as axial mesoderm, paramesoderm, intermediate mesoderm, and lateral plate mesoderm. And we are now working on the origination of the axial mesoderm in the lab. And to our more interesting, because we focused on the notochord, we know that notochord is a signaling center or organizer in the early human embryonic development that can secrete the sonic Hae-Ock signaling to pattern the embryo axis. And to our surprise, this story tells us there are another organizer might exist at the tail part of the human embryos, which is the collecting stalk, and the collecting stalk will finally develop into the umbilical cord at later stage of human embryonic development. So we verified the organizer features of this collecting stalk, and we hypothesized that the PGC and also a lot of hematogenesis happened because of these tail organizers. And more interestingly, we are now working on an earlier stage, human gastrula, which is the Carnegie stage 7. We focused our study on an even more mysterious structure, which is the node. And we of course, identified those mysterious structure nodes. And as you can see that it locates in the middle, very middle of the human embryos, and also we hypothesized that all the notochord cells can actually comes from this amazing node. And of course, we identified another cell type in the human embryos, because we all know that in the mouse, the determinant of the AP axis is the AVE, anterior visceral endoderm, and we finally identified another AVE, which we called anterior hypoblast in the human embryos. So because of the time limitation, my story will stop here. However, However, I think the interesting story of the human embryonic development still continues in the lab. So finally, I would like to thank Professor Hongmei Wang, who is the guide to lead me to the very amazing world of human placenta development and also human embryonic development. And also, I would like to thank my amazing colleagues, Wang Meijiao, who is working in CAS now, and thanks for him to guide us to the amazing world of all the computer science, and also my gradually growing talented students in BIT. And thank you very much.
Thank you, Dr. Xiao. Is there any pressing questions?
Yes.
That is such an incredible resource to go so early in human development. And I'm really curious to know if it's feasible to compare to primates at those stages, and if you can see any differences.
The primates, you mean monkey, like
Yeah.
yeah, when I compared our results to monkeys, they are quite conserved. Like the monkey also have lotocord and also node. But I think we are also very curious about whether those anxious determinants exist in other species, such as rabbit, such as chicken, and we are, of course, now we are using 10 species to do this comparison. Thank you very much.
Fantastic.
Yeah.
I have a question here. Did you also observe the axis of the definitive endoderm?
Definitive endoderm?
Yes.
The axis of the definitive endoderm in humans?
Yes.
Yes, I think for the definitive endoderm, you mean the axis? Because I think the axis is already determined by the AVEs or anterior hypoblast and also the lotocord. So what do you mean by the axis of the definitive endoderm?
It's also because of the contribution of the extraembryonic endoderm to the primitive gut. So I'm wondering whether in the definitive endoderms there also some kind of convergence you can observe in the human.
Ah, like him. Okay. Yeah, because you are working in mouse, right?
Yes, yes.
Yeah,
yes.
because for the mouse, I mean the definitive endoderm and also extraembryonic endoderm is quite different from the humans, because like the humans, the yolk sac is a big bubble in front of the embryos, and the mouse is totally wrapped by the yolk sac. So I think it's quite different, but still we know the conservation of the AVE and the anterior hypoblast. So yeah, thank you. Yeah,
hi.
Thank you.
Really wonderful. I love it. I have a few questions more about the placental side. So I know in vitro, people have tried to kind of mimic the stage of syncytial formation and try to study what are the genes that regulate the merging of to form the multinucleated cells. I'm wondering if you can also see that, since you have the different time points of the placental development.
Yeah, you mean in vitro or in vivo?
For your in vivo data, whether the in vitro is accurate or not.
Yeah, I like your question. Thank you very much. And because for the syncytial, because I think now, currently, all the in vitro model can only mimic the very early stage of the syncytial. And there is another syncytial that I was quite curious about, and I still don't have an answer, is the primitive syncytial during implantation. And our primitive, our very limited results suppose that those syncytial, they are actually not comes from the cell fusion, but comes from the endoreplication. So we are working now on the endoreplication of the primitive SDV syncytial in vitro now in the lab. Yeah, thank you.
Can I ask one more?
Yeah, of course.
I'm also curious about the yolk sac, because I think there's also some questions which are not well resolved regarding the migration of microglia and macrophages into the brain from the yolk sac.
Yeah.
Do you observe anything there that could give us hints, or is it not the right time frame?
Yeah, for the microglia, it's also interesting. The lab, we have a manuscript already under revision now because of the pre-gastrulation stage of human embryos. We hypothesized that all the primitive macrophage that actually comes from the extraembryonic mesoderm from hypoblast, and they will definitely migrate to the brain region to become the microglia. Hopefully, we can sell out this idea or hypothesis in the near future. Yeah, thank you very much. Yeah.
Right. So very good discussions. I think we should put our hand together to thank Dr. Xiao again. Thank you.
All right. Our second speaker is Jong-Eun Park from KAIST Korea, and he has been deeply embedded into the human cell LS network since the very beginning as a postdoc with Sarah Tackman. And today he's going to talk to you about liver aging process. There you go.
Yeah, this works. Okay. I'm Jong-Eun from KAIST. Nice to meet you all. And just a brief introduction about KAIST because many people ask me, are you in Seoul? Actually, we are not. We are a little bit southern part in Korea, like in the middle, Daejeon city. And my lab, named Single Cell Medical Genomics Lab, we want to focus all the aspects actually covered in Human Cell Atlas, like development and aging and disease. So for the developmental side, I have been worked in immune development, especially focusing on human thymus. And then we are also working on disease side, like inflammation and autoimmune side, with all the Korean heroes from SCADE project. I'm actually running a lab with one of them, Jung-Sung Lee, together. So I'm learning a lot from him. And we also are working on understanding tumor microenvironment, which we have done some of the pan-cancer addressing work. But today I'm going to focus on this aging microenvironment, which I believe it is all deeply intermingled, interconnected. But today I'm going to focus on one method that we developed to understand the somatic mosaicism, but at the transcriptome or epigenome level at mouse liver. So this one depicts our aging process from 23-year-old young lady to 100 centennial. And if you think about what's happening in between, it's like a 70 years period. So there's a lot of small things that is happening all the time. And then my question is, what happens at the beginning of the aging process? How can we define the first aging niche? And is it general in all cells? Is it happening to all the cells in our body? Or will it form a micro niche that we can specifically define, but it is difficult to detect because they are so rare? So that's the original question. And I'm putting this in this concept that usually when we think about somatic mosaicism, people usually talk about the genomic layer, the mutations and SNPs. But then this somatic mosaicism would link to the next layer of the mosaicism at the epigenetic layer and also the cell state or tissue architecture, which can be defined by the single cell and spatial transcriptomics. And one of the ideas that I got this from is my previous work on the thymus from Sarah's lab, where we have collected the thymus across fetal development to the aging, because thymus is not only an interesting immune organ required for the T-cell biogenesis, but it is an organ that undergoes really quick degeneration. So all of you have non-functioning thymus. So why this organ degenerates so fast? So that's kind of question fascinated me. So from this data collection, what we could see from the development and aging process, yes, there's a genes that goes down and goes up during aging process, like proliferation genes generally goes down, and senescence marker generally goes up. And there's a senescence-associated secretory phenotype factors like cytokines that usually is associated with the aging phenotype. And what we can see is it's actually all different. Their expression pattern is all different across different cell types. So this already shows that aging should happen in mosaicism, and different cell types are aging in different speed and in different manner. So I got really fascinated by this pattern, and I thought that when I do the public data or reanalysis of all the aging-related data, then I could see a clear pattern that I saw on thymus. But unexpectedly, it was not the case. Most of the aging-associated changes that we find in general single-cell data, it was very subtle. So this got me into the question that, are we capturing enough age-associated features during our single-cell or single-nuclei prep? And we all know that we have a bias in our single-cell prep. We know that some cells are easily die or get lost, or they are not easily get dissociated. And single-nuclei prep, it's kind of like general to every cell, but then you only sample the abundant cell type. you kind of still miss the rare cell type. And if you imagine the first aging niche, it would be really rare in our environment, in our organ, in most of the organ except for thymus, which is already degenerating. These small aging initial spots will be really rare, so it wouldn't be really captured with the single cell or single nuclei sequencing method. So I did this imaginary experiment that what we usually do is the single cell dissociation. So what we are dissociating is a healthy functioning tissue which are not that rigid, which haven't undergone too much fibrosis, so they're easy to dissociate and they are healthy, they are easy to survive. But then what if I just throw away all the dissociate single cells, but just collect the remnants of this single dissociation, first round of this dissociation, and enrich them, and then if I do the further dissociation of them into the single cell and look into them in deeper depth, what would we see? So that was the question, and then I decided to apply this onto the liver, because liver is the function organ that usually do not age. It is highly regenerative organ, but at the same time, it is known that it can undergo the progressive change in their function, undergoing steatosis and fibrosis, and eventually sometimes you can develop into cancer. So it is the organ that really reminds me of the somatic mosaicism. And from Peter Campbell's study, he beautifully showed that actually in human liver, there's a lot of genetic somatic mosaicism. So what we have done, we took the mouse liver from young and old mouse, and then we did a first round of digestion, dissociation, and then we put them onto the strainer. And then all the ones that has been dissociated in single cell at the first round of dissociation, we consider as a control sample, the usual single cell data that we would get. And then we took some other remnants from the strainer, and then we did a second round of digestion, and then we call that finishing the fibrotic niche sequencing, hoping that we are enriching for the fibrotic niche. And I'm going to argue that we are indeed enriching the fibrotic niche by showing you some data. So first of all, when you look at the strainer from the old and young mouse dissociation, as you can see, obviously, we are losing a lot of cells on the strainer. And if we quantify them, in young, by the way, YC is young control, YF, young finishing, old control, and old finishing. So you can now understand our nomenclature. And for example, in young sample, we usually have all the cells in control. They get easily dissociate. But in old, all the cells are left on the strainer, so they are difficult to dissociate. And when we do the proteomics out of the tissue remnants of the secondary digestion, we can see that they indeed have a different ECM proteins. So they have a different type of the matrix. What about single cell level? So we had generated a lot of single cell library multiple times to make it to make sure that it's reproducible. And what was interesting is if you look at this YCOC, the control digestion example, young and old looks quite similar, right? They don't show dramatic change. Whereas if you compare YF or OF, the finished condition, you can see that there's already dramatic change in cell proportion happening there. And then we asked, then in old finished condition or young finished condition, the cells that are enriching in our experiment, is it associated with the fibrosis? So we look into the human liver data, which has undergone the serious cirrhosis, and then we took the cell signature out of there, and then we mapped to our data, and then we can see that indeed the cell population that is being enriched in our data shows the high score of this, what is called the SCA associated signature for the macrophages and endothelial cells and mesenchymal cells. So we are indeed enriching the fibrotic niche related cells in here. And one very striking example here in liver, the liver is basically a filter organ. So you have a portal vein which accepts the nutrient-enriched blood from intestine, and it goes through the sinusoid for the filtering, and then after that it left the liver through the central vein. So what you see is that for the endothelial cell, you see this zonation pattern from portal endothelial cell to L-sac to central endothelial cell. But then we discovered this mysterious population we named the fibrotic EC. And then the reason why we call the fibrotic EC is that they are really niched in the old finished condition. If you look at the cell proportion there, the old finished condition, all the endothelial cells that are coming out belongs to this cluster. So what are they? We can see that they express a lot of inflammatory-related chemokines and cytokines. And also, when we kind of projected the senescence score onto them, we can see that they are really matching well with what we have defined from in vitro study, the common marker of the senescence, and also from many other like age-associated in vivo studies. So these endothelial cells are the ones that we usually think of as a culprit of the aging. And then when we projected them onto the like pan-organ endothelial cell atlas, what we could see was that these fibrotic EC, they have lost the liver identity, and they now look more similar to the intestine or muscle endothelial cells. So they are kind of trans-differentiating into the endothelial cell from other organs. And then another interesting case, for the fibroblast and mesenchymal cells, liver hepatic stellate cells are usually mesenchyme, and you have some fibroblast in portal vein area. So the yellow HSC and green portal fibroblasts are the ones that are previously known. But then from our finished data, we can see that very interesting new fibroblast populations are coming out. For example, SMOC1 expressing fibroblast and WIF1 expressing fibroblast, the purple one and red one there. And then the red ones are enriched in finished but in young sample. And the purple ones are enriched in finished but more in old samples. So there is a dynamic transition between this fibrous phenotype that often associated with the young fibrotic niche, and then it changed to the other sort of phenotype in old niche. And what is characterizing them? It's that the young niche, fibrotic niche enriched fibroblasts, they shows the high Wnt signaling response, expressing a lot of Wnt inhibitory ligands, whereas this purple one, old enriched ones, are expressing a lot of ECM proteins that should not be there in liver. They should be there in the cartilage or other stiff organ. It is now being expressed in liver fibroblast. And we tried to kind of validate this by looking into the in situ data. And what we could observe was that these markers, all these markers, co-localize around the portal vein, where the intestinal blood flows into the liver. So we believe that in natural aging condition, it's the portal area that is usually getting most of the age-associated damages. So we are getting to see this kind of enrichment. And we also did this kind of the mild fibrosis model, and we could see that these features are increasing upon fibrotic damage. So this is again showing that this is a population that is related to the initiation of the fibrosis process. And what was more interesting about these populations is that they're interacting with the immune cells. So these fibrotic niche associated fibroblasts, they express CCL19, so they can bring the T cells and B cells into the site. And the endothelial cells, the fibrotic EC express high level of PDL1, so they can give the exhaustion signal to the T cell. So what's happening to T cell? We can see that as the young liver gets older, what is called the CXCR6 positive tissue resident memory CD8 T cells, which are known to be kind of autoaggressive T cell in liver, which has been recently described. We can see that from as the liver become young to old, they obtain the PD-1 high phenotype. So they are getting exhausted in normal aged mouse without any cancer or infection going on there in just lab mouse. And this also accompanies with the changes in dendritic cells, and we could consistently see that the T cell co-stimulatory signal goes up high in finishing condition, the fibrotic niche, but we could also observe that this dendritic cell signaling is also going up high in young fibrotic niche, YF, compared to YC. So already in young, 6-month-old mouse, something is happening in liver in this specific niche that is kind of inducing T cell activation and finally leading to the T cell exhaustion. And we try to validate this finding by the spatial transcriptomics method. So first we collected a lot of liver visium data, so on the mouse liver. And then from the H&E, we could pinpoint the portal area, which we think that where the fibrotic niche is first initially formed in natural liver aging. And then what we could confirm is that indeed all these populations that we found to be enriched in fibrotic niche, they are more enriched in portal area than any others. So it's like a portal associated early fibrosis happening in natural aging. And then when we clustered these portal spots of the visium data onto the multiple single cell annotated features that we had defined, then we could see that there are portal visium spot that represents the homeostatic liver with the fatty acid metabolism and normal hepatocytes. But then there are other two clusters, one representing the inflammatory niche, enriched with the WIF1 fibroblast, whereas the other enriched with the fibrotic niche, and there the SMOC1, old associated fibroblast and fibrotic endothelial cells are enriched in this fibrotic niche. And what we could also see is that the hypoxic signature, the hypoxia, comes in this with this fibrotic portal niche. So to understand better what's happening in terms of the regulatory pathway here, we kind of combine with our finishing protocol with the atoxic. So we could niche these rare cells with the finishing approach by taking the cells from the strainer. Then we can subject them to the atoxic to see what kind of epigenetic profile they have. And as you can see here, when we compare young and old in from this atoxic approach, we can see that there are indeed some population that are enriched in old finishing, and some of them are matching to the corresponding population that we define from the single-cell RNA-seq approaches. And when we looked into the especially endothelial cell, we could pinpoint the chromatin profile of the fibrotic endothelial cells. And when we look into the genes that are open in this fibrotic endothelial cells specifically, we could see indeed the signature of the hypoxia, the HIF1 regulation, and also KF4-based reprogramming feature is niched in this fibrotic endothelial cell, which might be responsible for their transdifferentiation into other other organ endothelial cells. And finally, we also try to perform the stereosig, which offers a much better resolution. And we could find the kind of virtue of doing the finishing to identify exactly the cell type that is niched in this niche. Because we know exact single cell profile, we could map them onto this stereosig liver data. And this red and green one shows that these fibrotic endothelial cells and old niched fibroblasts are indeed colocalizing near the large vessel, and we can see the PD-1 high TRMs are indeed sitting on that same niche. So the conclusion of today's talk is that by performing this quasi-spatial method to niche the specific neighborhood cells based on their physical properties, in this case the resistance to the enzyme digestion, we could apply the multiomics approach like single-cell RNA-seq or single-nucleus atoxic or proteomics to understand this niche, and we could actually identify in niche the early initial rare fibrosis-associated states from the normal aging liver. And in the end, I think we can utilize this to understand the early dynamics of the processes that can induce the physical changes in tissue environment. So we use the enzymatic digestion as a kind of a physical property, but you can imagine any kind of the physical property to enrich the chunk of the tissue, and then you can dissociate them and then perform the whatever analysis that you would like to see. And next thing that we want to do is we want to link this with the genome, the spatial genomics approach, because now it's known that the normal liver undergoes the somatic mosaicism, the somatic evolution of their genome. So we are wondering whether this kind of niche is associated with a change in the genomes. So that's the end of my talk, and today's topic has been performed by Kwon-Yong Tak and Myung-Sun Park, the two talented graduate students, in collaboration with the Professor Chun-Ha Kim's lab in KRIP, KAIST, the Aging Research Lab, with the Jooyeon Kim, another graduate student. And the thymus work, I thank a lot of people from Sarah's lab and Hanipa's lab for that amazing research. So thank you very much, and I'll take questions.
Beautiful data, Professor Park. I have a pressing question. So how does a finishing actually preserve the old fibrotic cells and therefore allow you to study the fibrotic niche?
Yeah, actually it's very simple logic that even the minute fibrosis, they would be more resistant to the collagenase digestion. So if we do the simple collagenase digestion, then those tissue will usually live on the strainer because they don't get dissociated into single cell. So if I take those tissue chunks and then do the analysis, that's the concept of finishing, then we begin to see very new interesting world. So that's the main concept, yeah.
I see, thanks.
Hi, Jung. It's a very nice talk here. Amazing talk, yeah. So I have one small question. So regarding the T cells interaction with those fibroblasts, have you checked the TCR, whether they are antigen specific or it's just bystander activation?
Yes, we are working on that now, and with some other models, we are trying to understand, and we do see some clonal expansion there. So there might be some antigen involved in it, but we do not rule out the potential of the bystander activation. There could be both of them, yeah.
So what causes these nodules to form? And you mentioned genetics, so you plan to do genome sequencing. But
we just done our genome sequencing. We are looking at the result now. But then what is the cause? That is a very interesting question. And we are trying multiple stimuli right now, which should be associated with the portal veins, coming from portal vein. This must be something. And I think it can be diet. It can be some microbiome that is still left in the lab mouse, or it can be just a normal homeostatic process in our body. So all those possibilities are possible. So I think now we have an option to just see the result, outcome of those stimuli, so we can kind of give the stimuli and see what happens with this fibrotic niche. So that's our next plan.
Have you tried in other organs? Do you also see cells left in the strainer?
We try some other organs, and then we see another interesting world from there, but it is not always necessarily associated with the fibrotic niche. So I would say here, what we are doing is the enzymatic digestion resistance-based enrichment, and that can be depending on different organ situation. That can be fibrotic niche, that can be some other interesting structure that are there. So I feel like it can be generalized as a protocol that is looking into the physical tissue of the property of the tissue.
Yeah. Great work. Yeah.
Right, I got a question for you, sir. It's nice work. So I got a question regarding mesenchymal cells there. So have you seen, you observed the mesenchymal cell contributed in the ECM deposition, which caused the fibrosis. Have you somehow observed any mesenchymal cells contributed in the ECM degradation, or does it express any ECM regulators to reverse this?
And
reverse this, or any parasites which nearby, especially in liver, to reverse this process.
That's a very interesting question.
In your single cell data.
Yeah, I feel like I have never focused on that question, so I'm going to go back and look into that signature. But I also need to look into the dynamics. So maybe we need some more data in terms of giving them an insert and then generating the dynamic data. Yeah.
Thank you.
The thymus is aging, or the programmed involutioning, like involuting. So I don't agree that the thymus is aging. Possibly they are programmed involution.
Yes, it's a program. But I think, how can we define the programmed and non-programmed? Because usually it's a mixture of the environmental stimuli and the genetics that the organ kind of deteriorates in different speed, and also in different mosaicism. So I would say it's really difficult to define what aging is, but we can literally say some chronological change in the tissue structure mosaicism. I think that's a neutral way of referring to this at the moment. Yeah.
I have a quick technical question. It's just, have you evaluated the dissociation stress created by the second
Very important question. And we do see more dissociation stress associated with the second digestive sample, but this dissociation stress has been defined in multiple studies. So we are kind of filtering them out even beginning from the experiment analysis. So we excluded all the dissociation associated bias gene sets starting from at the very beginning of our analysis, so that they don't kind of affect our downstream analysis. But that's very relevant point, I think. Yeah.
All right. Thank you so much, Professor Park. Thank you. So with that, it's really our great honor to have Professor Katie Cheah. So Katie, she's an emeritus professor at the Hong Kong University. She's also an elected member of EMBO. So today, Katie is going to share with us the understanding of the development and degenerations of the human invertebral disc. So Katie, when you're ready.
Thank you. Thank you very much. And I also want to thank Dr. Xiao for a lovely talk, and actually an introduction to the first part of my talk. Because it's very relevant, the notochord is very relevant to the intervertebral disc. So why are we interested in the intervertebral disc? And that is because, in fact, a survey of the kind of disorders that have the greatest impact on the quality of life of humans is actually, number one, is low back pain. And the reason for low back pain is because you have degeneration of the disc. As you can see in the MRI there, the degenerated disc, the black portion is your degenerated intervertebral disc, and in particular, the nucleus pulposus, because the nucleus pulposus is the core of the intervertebral disc as a hydrated tissue and provides functional support and shock absorption throughout our daily lives. But in fact, the intervertebral disc and the nucleus pulposus in particular receives enormous amounts of stresses throughout life, both mechanical stress, hypoxic stress, because there's no blood vessels there, nutritional stress, and lots of stress, basically. And imagine it's got to survive throughout. So in my talk today, I'm going to talk a little bit about the lineage origin of the nucleus pulposus cells using mouse as the model, because that's the only one we can do lineage tracing on. But very importantly, the human is actually our target to understand, and to understand the nucleus pulposus cells. And so I want to show you some of the studies we've done in comparing mouse and human to see to what degree we can use the mouse to learn something in human. And then talk about the changes in the heterogeneity in the nucleus pulposus during the degeneration of the human disc, and finding potential progenitors if I have time. So as mentioned by Dr. Xiao, the notochord is a very critical, very early embryonic structure that forms and patterns the surrounding tissue. And we've learned a lot about the function of the notochord from studying the mouse and chickens and other species, but we know very little about the notochord in human. But basically, very early on, the notochord is a rod-like structure which later on becomes segmented to form the intervertebral disc. And some years ago, we and others have shown that the notochord is the precursor for all the cells in the adult nucleus pulposus. This is done by lineage tracing, and so you just see where the green cells are. And even at 1 year or 2 years old, in the mouse nucleus pulposus, you can see green cells, suggesting that they originated from the embryonic notochord. So what happens in human? We assume that similar events happen in human, but one thing we do notice is that in humans, the nucleus pulposus changes in its cellularity and in the kinds of cells that you see within the NP. And so this is a sign of degeneration. And very interestingly, in humans, these notochordal-like cells, because they have features of a notochord cell like vacuoles, they disappear fairly early in humans. You can see that by the time you're 6 or 10 years old in human, you begin to lose them. But in the mouse, you never really lose them until very late. So the question is whether these notochordal cells are important precursors or progenitors that might be important for the intervertebral disc. And so therefore, we want to ask the question about the dynamics of the cellular heterogeneity in the nucleus pulposus during development and degeneration, in particular, to obtain some information so that we can try and develop in vitro protocols to generate human nucleus pulposus cells in the dish. And so, for the sake of time, I don't want to go into this in too much detail, but this is a comparison of the different stages in development in human and mouse that you find that the process, at least the appearance of the notochord, is conserved between human and mouse. And by comparing the single-cell transcriptomes in a human notochord from the published data and also some of our own data of different ages of development, the earliest being at CS7, and then and the latest for us in the fetal stage is at week 17 in the nucleus pulposus from human embryos. And so what we did was to actually just compare, look to identify the notochordal cluster from this, and you can see from the red boxes that actually the notochordal cluster is actually very small in those data sets. And we use actually, so this is very important. In the beginning of this conference, it was talked about annotation. So we need to know how to identify these cells. And so the annotation mostly we use are insights that are taken from studying organisms like the mouse, because we know very little about human notochord development. And if you look at the single-cell analysis, we can then see that there are changes in the proportions of the different cell types. I don't have time to go into the details, but you can see that basically from the arrow, we see that from in that direction, the downward arrow, it seems that from the analysis one would predict that this is the direction of changes in the cell signatures as the embryo, human embryo develops. And this is just some of the differentially expressed genes that we used to come up with that conclusion. So I mentioned just now that we wanted to have information as to how to generate nucleus pulposus cells in a dish. And so the other important analysis that we have done is to analyze the different kinds of enrichment for pathways during that process in order to help us guide the in vitro differentiation. And so that information has been very valuable by looking at the analysis in the human transcriptome, and also by comparing it with the single-cell transcriptomes in the mouse notochord lineage. We wanted to see whether or not how was the degree of heterogeneity happening, because there's much more data available in the mouse than in the human. And if you can see, just focus on the red boxes, it seems that there is some degree of similarity in the differentially expressed genes at different stages of development, embryonic development in mouse and human, but it's not 100% identical. I said that I wanted to talk about the cellular composition of the nucleus pulposus during aging and degeneration. I've just talked about development. And if you look at the degenerated nucleus pulposus in humans, you see that there's a lot of signs of other cell types, myofibroblasts, macrophages, and signs of fibrosis. So therefore, it is important to actually analyze the cellular heterogeneity in the human nucleus pulposus across the ages which we have. And we have now made use of our own patient material and overlapped it together with the non-degenerated disc in order to find the differentially expressed genes. And interestingly, we do find that there are signatures in the younger NP of notochordal-like cells, which we call NCLs, but other cell types as well, including fibroblasts. And so if we compare the molecular signatures of the human and the mouse NP, we do find some similarities, but not a huge amount. And if we make the projection from the mouse NPCs to the human NPCs, we see there is perhaps some indication that the nucleus, the notochordal-like cells might be going towards a more one of the other kinds of cells that you see in the chondrocyte-like cells you see in the NP. I think I have 1 minute left. And so I just, in this 1 minute, would like to tell you that amongst other things, it is important to identify progenitors in the disc, even from young and older discs, because they may predict. And so we have searched for progenitor signatures and managed to find one gene that seems to mark a set of cells that can actually are expressed in human and in the mouse developing new intervertebral discs, and they disappear during degeneration. And this is published work now, so I don't really want to go into it, but basically in this paper, we demonstrate that these cells actually can contribute to all the cells in the nucleus pulposus, and so must be a progenitor. And the question is if these cells exist in humans. And so they actually disappear. So many questions actually remain to be answered. So what happens to the notochordal-like cells during fibrosis in degeneration? I said that you could see more fibroblasts. And so we have tantalizing evidence in the mouse where we induce degeneration that the notochordal-like cells actually do, some of them can adopt some fibroblast-like features, which would be the feature of fibrosis. And so we have a model for this degeneration, starting from the early notochordal-like cell, which is like the early embryonic, and it goes through a whole series during life of having received huge stress factors which might in the end induce them to undergo a partial EMT, change their extracellular matrix synthesis and become fibroblasts-like and leading to degeneration. So there are many limitations and challenges in this and in embarking on this study. First of all is the samples, the availability of human fetal samples and controls for disease samples, and very importantly for the disease samples, the phenotyping has to be extremely carefully done. We need to have controls for these. The annotation of the cell populations that we see, that's very challenging because we don't have any standards for human. The sensitivity and the validation, do we actually identify all the genes? Some transcription factors may be present at very low levels, so we can't detect them, and we would need to validate what we find. And importantly, we need the spatial information, the cell-cell neighborhood relationships and receptor ligand changes that occur, because you can see in the human NP is a very large tissue. And so I guess I don't need to persuade you that we need a human cell atlas of the nucleus pulposus across the ages. And so I want to thank the many people who have contributed to this work, which is on this slide. And if you would just bear with me to have one advert, please come to the Hong Kong again, if you're not from Hong Kong, for the ISSCR meeting, which will be held in June next year. And there are many opportunities for presentation and abstracts. Many abstracts can be chosen for talks. And for the Hong Kong residents, young investigators, there are special grants for you. So please apply. The deadline is December the 17th, so that's very soon. And finally, we will have a pre-symposium here, which is a public outreach symposium, so please come to that as well on June the 10th. Thank you very much, and sorry for going over time.
Thank you, Cassie, for the very insightful talk. Let's open to questions. Okay. Maybe I'll start first. So you mentioned about that humans lose notochordal cells fairly early in life, whereas mice, they still maintain those cells. Is it because we're bipedal, where we walk on legs and 2 legs, and they walk on 4 legs?
It might be, because it's very intriguing that if you look at a wild-type mouse and you look in the tail, there are a few vertebrae where you actually see dysdegeneration. And I very simplistically put it down to the fact that mice stand on their tails when they're feeding. And so it seems to coincide with those particular discs that are where the mouse is standing. And so it might be a function of the mechanical load. And in fact, as I said, the disc is actually subjected to a lot of stresses, not only biomechanical, but hypoxic, inflammation, everything, you know.
Great, thank you. More? Okay, all right, I guess in the interest of time, let's thank all the speakers for the session. So we'll have a break. And if you have not loaded your slides, please do now.
Also, because we've gone quite a bit over time, to make sure that everyone can catch the shuttle, let's shorten the break a little bit. So please come back later for the next session. Thank you. They'll ring the bell. They'll ring the bell.
Hello, everyone. Welcome back from refreshment break. I'm Stephanie from the School of Biomedical Sciences, Faculty of Medicine at the University of Hong Kong. So, hello. I'm a cancer biologist, and I'm happy to be chairing this session on cancer with my co-chair, Yuki Okata, from the RIKEN Center for Integrative Medical Sciences. So we've got 3 speakers in this session today, and our first speaker is Professor Alfred Cheng from the School of Biomedical Sciences and the Chinese University of Hong Kong. Alfred is a longtime friend and collaborator. He has expertise and long-term interest in epigenetics and also more recently precision immunotherapy in hepatocellular carcinoma. So I'll pass it to you, Alfred. Thank you.
Hello, good afternoon. Thank you for coming back from refreshments, and the organizers is very nice giving me the opportunity to share our work with you. Thank you, Stephanie, for invitation. I will have a story of liver cancer immunotherapies, data derived from single-cell sequencing. For those working on cancer, you will know that the immune checkpoint have changed the paradigm of treatment, and now become the standard of care of treatment. You probably can find your cancer of interest in this graph, showing a very interesting data that the higher the mutation of that particular type, the mutation burden higher, the response from the PD-1, PD-L1 would be higher. The reason would be probably due to the new antigen derived from the cancer mutation, and that will attract the immune cells protected from immunotherapy, leading to response. So for my interest, the HCC having very low or modest burden, the response rate is also very low. So what can we do to help the patients? This is the landscape of the immunotherapy treatment for HCC along about this 17 years, from tyrosine kinase inhibitors, the targeted therapy, to immunotherapy. So you can see that the field is developing very intensively. But to scale down all the studies, the bottom line is even the best treatment, the response is still only about 30%, meaning that 7 out of 10 patients still cannot be benefits. And in addition to the no mutation, the liver cancer transcriptome also cause problem. We can see in pancreatic cancer or other cancer, which is immune cold, liver cancer also is characterized by a lot of immunosuppressive cells and lack of T cells. And we have previously reported that some of the cancer signaling is involved in remodeling the tumor immune environment. We have a previous talk on fibrosis, and we also find that fibrosis in the surrounding environment also affects immunotherapy. And interestingly, it can be remodeled by epigenetics. For liver cancer, there are different etiology. So hepatitis B virus is in this locality, but in obesity or this kind of fatty liver associated cancer, the response is also different. So we have also been doing some work on that. But the current status is about the adaptation of the immunotherapy, meaning that if the patient already treated by immunotherapy, if they are not responsive, what can we do? What is the adaptation? So in addressing this question, we need to have our own mouse model, because most of the mouse model in the academic is responsive to immunotherapy. So we are doing kind of a in vitro selection of immunotherapy, and then let the tumor cells with the immune checkpoint blockade in the host environment. And after several rounds of this selection, we can generate immunotherapy resistance tumor. So the yellow is the parental sensitive cells. They are regressed after anti-PD-1 or anti-PD-1 treatment. But after this selection, we can make immunotherapy resistant tumor. So you can see very large tumor, and they are no longer responsive. So having this model, we find that the profile is quite similar to the patients in the immune exclusion. So in the resistance tumor, there are more immunosuppressive myeloid cells like MDSC or macrophage. And the T cells is characterized with exhaustion and lack of cytotoxic cytokines. So we have this kind of good mouse model. We also need to have patient sample, and in collaboration with Stephen Cheng, a clinical oncologist in our university, we also have a trial with Pembrol, PD-1, before treatment and after treatment, and then have around 20-something patients responding or non-responding. We perform the single-cell RNA sequencing, and after this, about 300,000 cells, we can characterize different cellular components. And the first part is about the PPAR gamma is activated or induced in the tumor cells after the immunotherapy treatment. You can see in the blue bar, the patients are non-responsive to immunotherapy. They are activated after treatment, and they are adapting to the environment, adapting to the immune checkpoint. And in this story, published last year, we find that immunotherapy actually induces adaptation to the tumor cell. PPAR gamma is induced, in which it is a transcription factor activating genes like VEGF, which is remodeling the environment. The MDSC would be increased, T cell would be exhausted. So for this story, we need to have a preventive kind of a treatment. We need to inhibit the PPAR gamma together with the PD-1 or PD-L1 so that the tumor environments cannot be remodeled. So this study demonstrates the importance of the new mechanism about how the current IMBREF 1550 clinical trials is working, and this immuno checkpoint blockade together with PPAR gamma can be further developed, and it is a new strategy. And so this is a first study showing that the mouse model and also the patient sample can be combined together. But the PPAR gamma antagonist is still developing. We hope that we can have a drug in the for patients later on. But is there any drugs that can be used at this moment? So we go back to look at the patient sample we have. Because we have previously shown that the HDAC, histone deacetylase, is contributing to immunotherapy resistance in HCC. We want to know, but this study is on the NASH HCC. But in our locality, most of them is HPV related. We want to search all the HDAC in our human genome and ask which isoform would be associated with poor response, poor survival after immunotherapy treatment. And we find out that among the 20, the blue one, HDAC 1, 2, 3 are those, if they have high expression, the patient will survive poorly after immunotherapy treatment. And they are overexpressed in the non-responder tumor cells compared to the responder. And more interestingly, when the tumor have this high HDAC Cas1 isoform expression, they are associated with deficient interferon signaling, which is actually very important signaling for the immunotherapy response. The tumor will be exposed to interferon gamma, which is transducing signal to activate the anti-proliferative effects, upregulation of the antigen presentation, or T-cell chemoattractants. So this pathway leads to tumor eradication. So the tumor cell adapt by mutate this pathway by genetic mutation. But there is not much data showing non-mutational regulation of this pathway. Because of this HDAC, which we think that maybe there will be epigenetic regulation of interferon signaling. And we need to have tools. And in the market, there's a HDAC inhibitors, class 1 HDAC. You can see the HDAC 1, 2, 3 are the targets. And the good thing is it is already in clinical trial, proved to be safe. So we get this of this drug. And then my postdoc, Yaling, have made use of the model we have generated, the resistant model. And first of all, we find that these HDAC are all upregulated in the resistant tumor. And you can see when we treat with the anti-PD-L1 or anti-PD-1, this tumor does not show any response. They are resistant. But the good thing is when we combine with this immunotherapy, with these class 1 HDAC inhibitors, you can see the tumor regress significantly. And when we profile the immune environment, we see the cytotoxic T cells, including the CD8, CD4, NK, or NKT. These cells are carrying out the anti-tumor action, and they are inversely correlated with the tumor burden. And they are also cytotoxic, And the survival-wise, the combination really can promote the better survival. The single treatment does not do much. Even though we have the cured rate, and then we challenge with the tumor, this tumor can spontaneously regress. So there's the epigenetic memory, meaning that this therapy is very useful. To understanding why, we perform the multiomics, single cell and single cell ATAC. We find out that in this single treatment, there will be not much change, but in combination, in this combo group, we see more infiltration of immune cells, similar to what we find in the flow, and then tumor become less, not that special, but we find that when we subcluster the tumors, there are subsets of tumor cell that is actually increased, whereas the other is reduced. So what are they? We find that when they are, this pathway of interferon signaling is the mainly enriched pathway. So previously, this pathway, when it is resistant, they are suppressed. But after this combination treatment, it can now be epigenetically reactivated. So this GC enrichment also find that the interferon gamma signaling is the top one reactivated pathway. All the genes involved in antigen presentation, the anti-proliferation, and so on become reactivated. Motif analysis from this single-cell multiomics also indicate the signaling transcription factor STAT, IRF is also activated. So with the help of Stephanie, we also have the spontaneous model. This combination therapy is also working very well. You can see significant reduction of tumor burden and also prolongation of the survival. So with this data, we think it is a good time to translate our findings. So there is a HDAC class 1 overexpressed HCC patients, and they are having poor survival, and we find out that they can reactivate the immune cycle, leading to changing the immune code to immune hot environment, and this epigenetically affects the key interferon signaling and also the apoptotic signaling. So this can serve as a very good epigenetic therapy, and so we go to Nick in Oxford, who previously provided drugs to us. And so we have a new clinical trial with Stephen. The patients is non-responsive HCC patients, and in the experimental arm, we treat with the epigenetic drugs plus the anti-PD-1, and then we are looking forward to the results compared to the standard of care. So with these patients, we also, like the first trial, we have pre-treatment biopsy and post-treatment biopsy. We are having the ongoing single-cell analysis and trying to see the biomarkers if there's a response. So with that, the take-home message of this short talk is that we find that integration between the mouse model and the patient sample would be very instrumental to identify new therapeutic pathway. In tumor cells, we find that there will be transcriptional regulators or epigenetic regulators, and some of them can be readily translated to a clinical scenario. And not only tumor cells, the tumor immune microenvironment, especially some kind of macrophage, may also be actionable and targetable. So with that, I would like to thank my lab and also Steffi, Xiyun, who is also in this audience, and my collaborators. With that, thank you very much.
Thank you, Alfred, for that fantastic talk. Questions?
Alfred, really great to see how combination therapies are coming out of this research. And also the TREM2 macrophages at the end from your COSMICS data, which, if I'm not mistaken, those are also the SPP1 expressing macrophages?
Right. This TREM2 macrophage is also positive in SPP1 as well in this subset of macrophage. Yes.
Okay, great. And so they show up, TREM2, SPP1 macrophages show up as a subtype in many different cancers, and in their co-occurrence, and they tend to be spatially co-localized with the malignant cells, I think, at least in some cancers. And that's, I guess, what you're seeing as well, right?
Yes, very interestingly, this TREM2 or SPP1 positive macrophage would be much more in terms of proportion in those immunotherapy resistant patients, much less in treatment naive or responsive patients. So they're really changing the environment. And what we find here is they can remodel the tumor cells and making them more immunosuppressive in terms of affecting the T cell and so on.
And are they secreting TGF-beta, or is it coming from the fibroblasts?
That's a very important question. We find that, interestingly, the myeloid cell, the macrophage, also may secrete the TGF-beta, forming a kind of a circuitry and a very self-reinforcing circuitry.
Okay, thanks.
Yeah, it's a good story about HDAC inhibitors of clinical trials. As congratulations. So I was wondering, is it possible that HDAC inhibitors in the changing of epigenetics of expression profile of not only the tumor cells and the other, immunocell, the also basal cell, the fibroblast of the,
Yes, thank you. This is a very important question. So the drug, because it is systemically administered, it will affect different cells. And in fact, what we find another key target cell would be the T cell. As you see, one of the data is the epigenetic memory that we think, because stop the treatment, the tumor or the host already have the memory, so that when you have the recharge of the tumor, it will regress spontaneously. And what we find that some specific T cell, CD4, they are highly expressing the HDAC8. When we inhibit that, there will be epigenetic memory inducing. So we have been doing that in a separate experiment.
How about the basal cells?
Basal cells.
Basal, blood cell, blood, angiogenesis cells.
We do not find HDAC8 or HDAC123 expression in the blood cell.
All right, thank you. In the interest of time, we'll move to the second speaker. Thank you, Alfred.
Thank you.
Okay, so it's my great pleasure to introduce the second speaker of the session, Hae-Ock. She's a principal guest at the Catholic University of Korea. Okay, please talk.
Okay, good afternoon. It's my great pleasure to share our work with you here. So I'm going to talk about lung cancer, focusing on two ongoing studies. So one is the characterization of tertiary lymphoid structures using spatial and single-cell RNA sequencing data, and second part is the use of B-cell receptor sequencing data to generate anti-tumor antibodies. So I had a really good introduction from the previous speakers. I'm going to talk about immune checkpoint inhibitor aspect. There are biomarkers that can predict positive response to immune checkpoint therapy. And the best known is the PD-L1 staining done by immunohistochemistry typically. And the second one is tumor mutational burden, which tells you how, what is the abundance of neoantigen, the presence of tumor-specific antigens, and third one is the presence of tertiary lymphoid structure. Tertiary lymphoid structure, TLS, is an ectopic lymphoid organs that develop in non-lymphoid tissues, such as a tumor. So classically, the immune response, if you have an antigen in tissues, dendritic cells are supposed to pick up the antigen. They migrate to the secondary lymphoid organ and present the antigen to like a naive CD4 T cells, activate CD4 T cells, CD8 cells, B cells there. So there you develop T cells, the differentiation, proliferation of T cells, and also memory B cells. Then they go back to the tissues and kill your tumor. That's the classical view. And these events may occur directly at the tumor sites within the TLS. So the typical structure of TLS, you would have a B cell core and surrounding T cells and dendritic cell network. There may be other cell types involved, but that's the basic structure. And for many tumor tissues, the presence of TLS has been shown. The typical marker would be T cell markers and B cell markers, and you can use more of the molecular features of the TLS using a set of chemokines such as CXCL13, CCL19, etc. So using single-cell RNA sequencing data from this landscape studies, you can have a hint whether you have those type of cells, but you don't know whether they are in the TLS. There are multiple phenotype T cells there. And for B cells, for the lung cancer, there are a lot of follicular B cells are detected, a few germinal center B cell phenotype, and also plasma cells. And for the dendritic cells, there have been many different phenotype dendritic cells, and you can see the activated dendritic cells expressing CCR7, LAMP3. That's frequently found in that TLS area. And they express high levels of CD86, 83, 80, the co-stimulatory molecules. So to detect this TLS structure, we have to look at the tumor microenvironment in spatial context. So we have set up the project that we have about 19 Visium data and also produced 14 Xenium data with 476 genes for the lung cancer and immune cell panels. We also have some clinical data like a PD-L1 expression, tumor mutational burden measured by whole exome sequencing. We have adenocarcinoma, squamous cell carcinoma. I will not talk about in detail for those different types of tumor, but we are looking at like a general landscape using our data set. So we have generated coordinated single-cell RNA sequencing data. Instead of a typical subclustering that we have done a lot for the heterogeneity measures, we try to find as many different phenotype cells. We have done the one-step clustering because we are going to use this information for the Visium deconvolution and Xenium label transfer. So we thought it would be a better strategy to the one-step clustering. For the Visium data, so we incorporated all the data together for 19 patients. We have done the clustering analysis. So we have followed the SORA pipeline for this clustering analysis. And you could see nice areas like enriched with more of tumor cells, myeloid T cells, etc. And we focused on one cluster, that's cluster 10, which shows not only the T cell markers, but B cell markers and chemokine CXCL13 and CCL19. That's a typical chemokine signature for TLS. And when we map this clustering results onto the tissues, you'd see the yellow spots. That's the cluster. And you'd see for some patient tissues, you'd have this aggregated structure for the clusters. For some, you rarely find those structures, some showing scattered patterns. So using the differentially expressed genes for this cluster 10, we reconstructed gene network, which shows like typical TLS network, T cell network, B cell network, their interacting network, and also two other cell types. And using the DEGs, we could redefine the Visium regions that, of course, we use the DEGs, so they will define it. And we have actually tried different public signatures for TLS. For some signatures, they uses a lot of different genes. Actually, they don't do quite well. But for smaller signatures, many public signature to define the Visium data we have. We went further and we tried deconvolution of those spots. You can see the resolution got better. Of course, we don't know whether this is truth or not, so we have done the Xenium data as well. For the Xenium data, so we have generated matching data for 14 data set. For the cell type annotation, we have used both unsupervised clustering with 476 genes and also label transfer from single-cell RNA sequencing data, and you can see the nice correlation. And you would see for some cluster, you would see there's a tumor label and also other cell type label. It could be like segmentation issues, but actually you can see the data interpreted as a junctional signature. So I think rather than worrying about this segmentation issues, it may be good to use that segmentation issue data as a cell-cell interaction data. So I would expect the bioinformaticians see that as a good opportunity to do that. This is mapping integrated spatial clusters on the Zhenyu data. So this is, again, integrated clusters, and I will just focus on one sample, one on the bottom from 1 to 3. Third, so this is a comparison of the Visium cluster, Zhenyu deconvolution, and Zhenyu label transfer data. First, you will see a better resolution data with the Zhenyu, but you can also see that Visium deconvolution does really good job. So we've actually used the matching single-cell RNA sequencing data, so that's a bit better than we use the public single-cell RNA sequencing data. But I think if we use enough of single-cell data, we may do it without matching the patient data. And this is a demonstration of the TLS structures. So one on the left is public data from 10X site, human lymph node showing the B-cell follicle that can develop into germinal center. The one thing that I want to point is that for the public data set, we could use it as a good control, but they don't have markers we want to see for the TLS. So they are missing like a BCL6, so we can look at germinal center. Our samples, we use the BCL6 for the yellow dots, so we see a nice germinal center area surrounded by B cell, T cell, and dendritic cell network. So we wanted to use this tertiary lymphoid structure signature to something, link it to the clinical annotation, but we don't have that. So for our spatial data, it's only a resection of the early stage cancer, some stage 3, but mostly resectable cancers. So we don't have clinical information, any therapeutic response data. So to link that, we have to translate this Visium data or single-cell data into bulk data so we can measure the abundance of a TLS. So we use the DEGs to score bulk RNA sequencing data, and that data shows a good correlation with the Visium TLS area. So this is produced from our data set, one with the Visium data and bulk RNA sequencing. So we applied this signature to a public data set from Roche, and they show a predictive value for the immunotherapy. So it's including all data set, and one only including the immunotherapy data. So the TLS signature can be predictive. So we are analyzing our spatial data in addition to TLS. So there are many features, but I wanted to show one aspect of things that you can get from your spatial data. And the second part, I'll be very brief. So this is a use of B-cell receptor sequences. It's my hobby project, or could be a dumb project. You will see why. So this one started from our animal study. We are studying animal models to look at the mechanisms of immune checkpoint therapies. And one of the study material was anti-CTLA-4 treatment. So after the treatment, the tumors regress, and you'll see the red, that's the tumor. Tumor regress, and there's a lot of immune cells are expanding. It happens for the T cells. So the blue part is a single clone that we find. So there's like a recruitment and also expansion for the orange colors after CTLA-4 treatment. In this mouse, you also see an expansion of B cells. And these B cells show a different phenotype, many follicular B cells, plasma cells, and small number of germinal center cells. And if we stain the tissues, not all mice, but some mice show a good area of B cell and T cell co-presence. So we decided that if this is the area where your antigen-specific B cells and T cells differentiate, they have to be tumor-specific. So we went ahead and generated the recombinant antibodies using the B-cell receptor sequences. So we selected the sequences where we see the in situ differentiation. So the red marks one specific clones that's found in follicular B cells as well as in plasma cells or like an expansion in germinal centers. So we suspect they are tumor reactive. And we generated, stained the cells we have used for the mouse model. So it's a transplant model. So they are reactive to CT26 cell line, but the reactivity is quite low. We could barely see the shift. But for the tumor cells, we have actually injected the mouse and then got back from the mouse. They show a quite good reactivity. And we treated the antibody back to the mice, and some of the antibodies, they induced the tumor regression. So in tumor volume and tumor weight, there are some antibodies that was reactive to the tumor cell line. Actually, they make the tumor regress. So can we find the tumor-specific antibody in human samples? So we have followed the similar pipeline. So we selected B cells showing in situ differentiation. So take clonotypes that is found in follicular B cells as well as plasma cells or ones proliferating in the germinal centers. So you can see they are signatures. Particular clone showing GC B cells and different cell barcodes showing plasma cell signatures. So we generated antibody, tested whether they are reactive to tumors, but not reactive to the normal tissues. This is a pooled antibody, I think 5 or 6. So in general, maybe they may be reactive. They should not reactive to the normal cells. So we have used the blood cells. Compared to the positive samples, which is stained CD11B, all the antibody clones that we have made did not react to the normal blood cells. So we tried to screen tumor tissues, but it was really difficult. We have no matching tumor tissues, so different patient, it was really difficult to find. And once we find the positive samples, how are we gonna find the tumor antigen? It was really difficult task. But then I thought that if it's gonna be a general tumor antigen, we may find it in cell lines. So we screened different lung cancer cell lines. Again, cell lines did not show a good reactivity, maybe some very slight shift. Then we injected these cells into the mice, grow them in vivo, although it's mouse. Then we find some antibodies reactive to those cell lines. So actually, this is my last slide. So I told you that it's an ongoing study. And what we want to do is that whether we can find a tumor antigen from this dumb side of view. Because it's going to be much easier if we go from the tumor antigen and generate antibodies. This is a finding antibody. We have no idea what the antigen is. So we may end up finding it's like a EPCAM or some common antigens, but it's a different angle at least. So we thought if we identify tumor antigen by this approach, and fortunately we have several antibodies that shows a good negative and positive peak. So it's gonna be relatively easier comparative analysis. We can look at the RNA-seq data, proteomic data, or glycoproteomic data after sorting those populations. So once we succeed in identifying the tumor antigen, we'll screen all of the antibodies that we have generated from the lung cancer patient. We also have followed this procedure in ovarian cancer, so we'll expand the study. So with that, I'd like to thank all my laboratory members, especially Hiram, Gunsop, Serim, 3 students involved in this project. Also my collaborator, especially Seung-Joon Kim from St. Mary's Hospital and Myung-Ju An from Samsung Medical Center. Thank you.
Thank you so much for great talk. So now it's open for discussion. So maybe because of the behind the schedule, but you definitely want your questions. Yep.
That was a great talk, and I love the thing at the end as well. for the antibody. But related to the TLSs, so if you're going to an immunology crowd, it's they have very specific requirements. So to actually call it a real TLS. So have you thought about how you're going to get it across the line to say that your signature really is identifying TLSs? Do you need to have antibody staining or pathologist or whatever?
Right. So actually, Xenium, you can see all the expression data-wise for the chemokines, all the necessary signatures for TLS. I think that's more complete, but I agree with you that we have to do the classical things. So we have leftover tissues matching frozen. So this is fresh frozen tissues. So we will do the staining once we select all the markers. I'm going to use TLS markers, but I haven't talked about it, but we found some interesting features for the macrophages. So once we set the markers, we'll do the protein staining for all those samples.
And I was just wondering, do you see any different distributions of IgA, IgHA1 and IgHG1? Because in ovarian we've seen quite different distributions.
For the lung cancer cell data, for the single cell data, we did not see a good separation of the IgA and IgG. I know that for like a colon cancer, you have a good separation of those as a clusters, but for at least our data set, they do not show a good separation of the IgG and IgA phenotype.
Thank you.
Okay, thank you again for a great talk. And let's go to the third speaker. Yep.
Thank you. So our third speaker is Dr. Hui-Mei La, who is currently a research fellow under the supervision of Sean Grimmond and Fernando Rosello at the University of Melbourne. And Mela will be talking to us about how she's using single-nucleus profiling and spatial transcriptomics to interrogate the pancreatic cancer tumor microenvironment. May please. Thank you.
Hi, everyone. Thanks for having me here today. It's my first time at HCA conference, and I've been enjoying it a lot. So for today's talk, I'll give you a quick overview of the project I'm working on, which involves pancreatic cancer. And because of the limited time, I'll just give you a quick story of how we have integrated spatial transcriptomics to help us interpret better our single-nuclear RNA-seq data, and combine how the both assay will give us better understanding of the tumor microenvironment. So this slide just to remind us how deadly pancreatic cancer is. And if you compare pancreatic cancer survival rate versus many other cancer, it's remained extremely low, like less than 10%. And they're quite ineffective to a lot of the treatment. So meaning that there's still a lot that we don't know about this disease. So I won't talk too much about the ICGC, but this is to give you a quick overview of where our sample come from. So it was part of the ICGC large cohort study where our group and others has performed a large-scale whole genome sequencing to derive important driver mutation and also subsequently the bulk RNA-seq to define the molecular subtype of pancreatic cancer. And from these well-characterized tumor, we select a subset of these samples for further assay like single-nuclear RNA-seq. And more recently, we also incorporate some additional sample, but from a different registry, so they're not matched, to perform spatial transcriptomics. And hopefully, when we combine these two assays, we can characterize better the cellular ecosystem, the stromal immune cell perturbation in the cancer setting. So just the overview of the method. I won't go too much detail into the single-nuclear RNA-seq because it's the standard workflow that everyone has been publishing. But I want to spend a bit more time discussing how we did our Xenium in situ gene expression. So for this, we actually profile 314 genes that we specifically curated based on the analysis from our single-nuclear RNA-seq data. And then we also verify with additional study with a very simple but specific goal is to, can we validate all the cell type that we see in the single cell data? And if we can, what are the spatial relationship between them? And the remaining step is pretty automated within the Xenium analyzer. And the output you can export for further downstream analysis. So after the pre-processing step, we did the manual annotation, and this is the atlas of the pancreatic cancer that we have. On the left, where the cell being color-coded, And the right is just to show you the heat map showing the marker gene that we define these major cell classes. And importantly, using the same marker gene, we can also define the concordant cell type interspatial data, of course, in the broader scale, but with the advantage of actually seeing them in the spatial context. And since we were talking about TOS, we actually have a little TOS here too. So after we got the data set, so we know that we have, now that we know we got the very high quality single nuclear data set and also the high resolution spatial transcriptomic, there's a lot of data that we can mine out of this, and we can start asking or addressing a lot of the outstanding biological questions. And one of the cell types that I want to discuss today is tumor-associated macrophage. So this macrophage is known to play a diverse pathological role in cancer, involving in like dysplasia, ECM remodeling, drug resistance, chronic inflammation. However, this population has been relatively understudied in the context of human pancreatic cancer, and also especially in, it wasn't well described as well in the recent single nuclear RNA-seq from pancreatic cancer. So we thought we would use our data set to characterize this population a bit better. So we use this, so from our single nuclear RNA-seq, we can clearly see a 4 robust subtype of tumor-associated macrophage across multiple patients. The first 2, as you're all familiar with, because they commonly detect in multiple cancer and also in pancreatic cancer as well, the SPP1 or the TREM2. They're known to be immunosuppressive, pro-angiogenic. And the second one is C1QC, so they often enrich in the complement pathway or pathway in phagocytosis. But the 2 additional subtypes that we detected is the OLR1, which is quite similar to SPP1. If you can see from the map there, they express they uniquely express the OLR1, which is another scavenger receptor. And we suspect that it also involved in immunosuppressive as well. And finally, as the name implied, MXC class 2 high, this is a population that express much higher level of MXC class 2 related gene, potentially involved in antigen presenting. So now that we know there's a 4 subtype, potentially subtype, we now turn into our spatial data to really see whether we can define them. So here I only focus on one sample, focusing on this small region here. And again, you can analyze them like the single cell data, and on the dot plot over here just to show you how confident we are in determining the cell identity of these population. And of course, now we can see where they really are at a very high resolution. Just to help you visualize a bit better, we can look at the region 1 and region 2. So region 1 is where you have a lot of SPP1, OLR1, that highlight in blue. It's like the previous mentioned, they seem to be associated with malignant cell. Whereas if you look at the C1QC, they were clustering on the far left corner, intermingled with many different cell types, and seemingly away from the malignant cell. And for that, we can perform the neighborhood enrichment analysis, which is a spatial statistic to determine whether any pair of cells are actually having any spatial relationship. And in agreement with what we just see, a malignant cell are often enriched with SPP1 or OLR1 tumor-associated macrophage, whereas the C1QC are often enriched with immune cell. Sorry. So we know that was just a representation of a small region. We then next asked, is it something that we can see consistently across multiple samples? So then we went back to each sample, so we surveyed the whole section instead of the small region. Then we identified all the cell types that we can, and we did that for all of them. And then we summarized them as a relative enrichment. And now we ask the question, within the malignant cell neighborhood, within the 50 micrometer, what are the cells in that neighborhood? And the one that have the highest one, of course the highest one is malignant cell, because malignant cell never present by themselves, so they always grow together. So then there's one, but the next one you can see is SPP1 or R1. And again, if you can compare to the C1QC, they're actually very, very low. And equally if you look at the immune cell, the lymphoid cell is also very low, indicating that C1QC and other lymphoid cells are actually very distant from the malignant cell. So in summary, through the combination of single nuclear RNA-seq and also the spatial data, we can start to form a hypothesis, working hypothesis that these SPP1 or R1, maybe they are pro-tumor growth because they was highly associated with malignant cell. They known to produce, based on the single nuclear RNA-seq, they produce all sorts of growth factor that has been shown in the preclinical mouse model to support cancer growth. And importantly, the C1QC we found in our data set suggests that they was never in the neighbor with the malignant cell. Instead, they're often found in tertiary lymphoid structure or the area with a lot of immune cell. And what I haven't shown you with MXE class 2 high tumor-associated macrophage. So this is an example where we'll need a better gene panel and also a bigger sample because we were only able to confidently identify this population in one sample where they seemingly also enriched with many other antigen-presenting cells like dendritic cell and C1QC. And for that, I would end my talk. I get a lot of support from my supervisor, Sean, and I would like to thank everyone else involved in the project. Thank you.
Thank you, May, for the very nice talk. Questions? Yes.
That was lovely. I'm glad I saw it this time. In your neighborhood analyses, are there any cell types that are depleted? So are they statistically depleted near tumor cells? So they never go near tumor cells?
So that's the C1QC and the lymphoid cell. They'll never need it. Yeah.
Glad to see that. Same question as before. We see this persistent pattern across many cancers.
Yes, exactly.
Great to see in pancreatic cancer, the dichotomy between SPP1 and C1QC in terms of their co-localization on the malignant cells, which brings up the question, the malignant cells must be secreting something that's creating that SPP1 phenotype, presumably, right? Does anyone know what that is?
So based on our analysis, because we all say that tumor-associated macrophage are immunosuppressive. So when I check the expression of the SPP1, they don't express any of the immune checkpoint inhibitor. They actually produce a lot of the cytokine and growth factor. That's why I feel they may be more involved in like pro-tumor growth environment, and maybe in turn, the malignant cell are actually the one that immunosuppressive.
My question is slightly different.
Oh, yeah.
It's very interesting, but why are the SPP1, why are the macrophages near the tumor? Why do they have the SPP1 phenotype? Are they responding to some signaling from the malignant cells?
This is something we're still figuring out because we cannot learn much from the mouse model as well. Like I tried to find some common thing, they're actually not much. Yeah.
Okay, thanks.
Actually, in relation to that question, I know that pancreatic cancer, one of the unique things is that it has different calf population that's worked out, right?
Yep.
So calves is also known to regulate macrophage and its differentiation, etc. So have you looked into, how your tumor-associated macrophages in that context?
At this stage, no, but based on the relationship, we know that it usually, if you're talking about the subset of calf, it's usually the myofibroblasts or the mycalf are the one that interacting more with the macrophage. Yes.
If no more questions, then we'll wrap up the session. Thank you again very much, Dr. Wang.
So, good afternoon. So I'm Michael Cheng from School of Public Health at HKU. I will be chairing with my partners on Dr. Paupin from Thailand on the immunology and infection sections. So for the first speaker, we'll have Dr. Jonathan from, and he will talk about the single cells and spatial cartogram of immune tissues. Give to you, John.
Thank you, Michael. Also like to take this opportunity to thank the organizing committee for your very kind invitations. So I must confess that I'm not an immunologist, neither am I an infectious disease scientist. I'm a stem cell biologist who works a lot with immunologists. So I'm going to give you a perspective on, as a stem cell genomic scientist, how what are some of the questions that we are trying to address together with our immunologist colleagues. First and foremost, our research interest is really, to understand how the combinatorial signals from epigenetic transcription factors and RNA modifications could coordinate to result in gene expressions during cell fate transitions and determinations in complex human tissues. Some of the earlier works that we have done, which we onboarded single-cell technologies, include trying to look at how during the reprogramming process cell fates change when you added in the 4 Yamanaka factors. So we harvested the cells from different time points. And then, so those were the days where you do not have a platform where you could look at within a single cell both RNA-seq and ATAC-seq. So we had to perform them separately and then computationally integrate them. So from this kind of work, we were able to identify the barriers of reprogramming like FOSL1, where if you overexpress with the Yamanaka factors, you could abrogate reprogramming, or the effectors proteins like TAT4, if you overexpress with the Yamanaka factors, it could improve dramatically the reprogramming efficiency. So we also look at human tissue. So this is in collaborations with clinicians who had access to the ocular samples. So this is something different from what was previously published, which were revolving around the retina. But in these particular studies, we also got the cornea tissues, we also got the sclera tissues, and we were comparing them to the zebrafish and the pig ocular samples. So perhaps of interest to these particular sessions, we identify quite a number of different subsets of immune cells, like the activated T cells, the monocytes as well. And some of these, we then went on to do computational studies, and they were actually tissue-resident immune cells that helps in the normal functions of the ocular systems. That was during the height of COVID-19. So we took the samples, data sets, and then we looked at the expressions of ACE and TMPR, both of which are very important for viral infections of the cells. And we found that, very interestingly, these conjunctiva cells within the corneal epithelium seems to express very high levels of these two proteins. So suggesting that, the advice from the public health to wear goggles for frontline health workers is justified, because the virus could use the ocular system as the reservoir before infecting the other persons. So very recently, Kiyu Hamashima and Gary Wong. So Gary Wong is a proud PhD graduate from Hong Kong University. So they got together to try to map the M6A. So we heard a lot about M6A modifications, profiling, but usually, it's in bulk. And there is a huge gap in the lack of single-cell M6A methodology and platform. So when we embarked on the projects, we found that there were two competing technologies. One of them is the SCM6A-seq, but that technology is really not high throughput. So you need to separate the cells into single cell, put them into single well of 384 well plates, and then profile them. It's not adaptable to the droplets technology, for example. The other technology is the SC-DART-seq, which requires genetic modifications. So that may work for transgenic mice if you want to profile tissue. Doesn't work if you want to collect primary human tissues for profiling. So Kiyu and Gary came together to invent and develop this technology known as single-nucleus M6A based on cut and tag. So I'm going to tell you a bit more on this technology. So basically, a primary antibodies against the M6A, secondary antibodies that carries the TM5. So when the reverse transcription takes place, the cDNA will then be targeted by the TM5, which will make a cut and insert a sequence, but not into the mRNA, into the cDNA. So we validated that technology. We found that 3' UTR, which is known to be enriched for M6A, were indeed so in our bulk sequencing. So we also sequentially reduced the number of cells in our M6A CT-seq down to 5K comparing to MeRIP-seq, and we found that in loci such as nano in SOX2 or in this case, I'm showing TET1 here, that they are highly enriched for M6A. So this is examples for NANOC and SOX2. So we then perform single-cell M6A technology, adapting it to the 10x chromium multiome, and that compared to the IgG pull-down, the pull-down with the M6A antibodies have elevated enrichments. And when we pseudo-bulk this single-cell M6A signal, indeed, we were able to identify previously known peaks at NANOC and SOX2. So to further validate, we compare to these wild-type mouse CSCs with the MET-L3 knockout mouse CSCs, and we found that indeed with the knockout cells, you do not see any M6A enrichment for genes that are known to be M6A modified like SOX2 and TET2, but not for RPL23 and PRDX1. So we then came up with version 2 of this technology. We found that version 1, although works very well for cell culture, doesn't work that well for tissues. So this is the version 2. We have submitted the manuscripts, not published yet, so I'm not going to review too much on the detail of the modifications to the protocol. Just that we have now perfected and to make it work on the attack kit, which seems to work very well, give us very high level of signal. In this case, I'm showing you examples of ZRP42, where we get a very high signal. So we are profiling different tissue types, including the embryo, from E7.5 to E12.5. We are also using it to profile another tissue type, which will be very relevant to these particular sessions, which is the thymus. All right, so Dr. Park had already given you some backgrounds on why is thymus important, and his earlier works on using single-cell profiling to look at thymic cells and the thymocytes in there. So thymus is where T cells are developed, and the intricate interactions between the T cell progenitor cells, which convert interactions with the thymic epithelial cells, which resulted in TCR rearrangement, and then they become double positive for CD4 and CD8 before they migrate into the medullary regions, and they further select by the medullary TAC cells before they mature into single positive CD4 or CD8. So from the perspective of a stem cell scientist, there are a few gaps that we wanted to address. So first and foremost, although embryonic stem cell has been very effective in differentiating into different cell types, but you realize that a lot of times when you differentiate them into T cell, you end up getting the CD8 positive cytotoxic T cell. So for very successful CAR-T, CD4 helper cells is also needed. So there seems to be a lack of knowledge of how to differentiate embryonic stem cell or iPS cells into CD4 helper cells. So that's one gap. The second gap is thymus will get aged very easily, known as involution, was mentioned just now in one of the Q&A. Thymus involutions resulted in the aging of the thymus, but embryonic stem cells, scientists have a lot of difficulties in differentiating these stem cells into epithelial cells, fully mature epithelial cells. So that's the second gap. And the third gap is, although embryonic stem cell has been able to make organoids for many different tissue types, but if you check out all the papers of human ES formations of the thymic organoids, you see that there isn't any structure. All right, so they simply get a globular lumps of cells that are somewhat functional, can cause T cell maturation, but there isn't a strict architecture that resembles that of the thymus. So because of that, we are interested in understanding the human thymus. So what we have done here was to use a very high resolution stereo-seq as well as stereo-site-seq for us to profile the thymus. So someone mentions about cell segmentation being a problem, so we think that technology like stereo-site-seq can partly ameliorate that issue. And we're also very interested in understanding the M6A modifications because M6A has been implicated in T cell activation and T cell development as well. So we have gotten samples from a fetus and pediatric, so I did not show here. We also gotten samples from adult age samples as well. So the project is driven by Cheng. She had the poster here. She presented yesterday. And Uma is the informaticians that work very closely with her. So we initially faced a lot of problems with stereosite-seq for the thymic samples, likely because of the probably the high levels of RNAs, probably the compact nature of the thymus. So if you look at pediatric samples 7 week, so that would be the typical samples or the data that we get after we send it for stereosec, about 200 to 300 genes per dot per locations. So lately we have been able to make changes to the protocol and sort of ramp up the resolution. So if you look at the pediatric 2-year samples, 5-year sample and so on, so these will be the kind of results that we are getting very lately. And that Professor Liu Bo-Xiang talked about it yesterday. So he was using data from site-seq of the mouse thymus. So we are aware of the publications by Chen Ao and his team in bioRxiv. So we work with them to onboard the stereosite-seq for the human thymus profiling. So perhaps not too unexpected, we see that the RNA and protein doesn't always align very well, which makes it justifiable that indeed for some of the very important markers, identifications of where identifications of particular cell types is critical, you should also have the protein profiling because the RNA profiling can be quite deceiving. So this is another example. So for example, cluster 11, 1, 11, and 14, we do not really see a lot of concurrence between the RNA expressions and the protein expressions, while in other clusters they do. This is the sort of methodologies that we are going to use here. So we manually select the spots using the EpCAM proteins, which is specific to the MTAG and the CTAG cells, and then use cell type predictions within each of these spots, and then spatial proximity predictions. So in Sarah's paper, I think they need to have a handle, which is to identify these HC, Hesse-Corpuscles, which is a very unique structures in the thymus. So they did H&E staining, and they then predict where, by the relative distance from the HC structure. So using our methods, we think that we will be able to simply from, this very high resolution transcriptomic data, be able to identify structure like the HC, which I will show later. So in the thymus, there's quite a lot of different types of tag cells, apart from the MTAG cell, which is at the medulla, the CTAG cells, which is at the cortical. There's also other classes of tag cells that express peripheral tissue transcription factors. So the rationale is that they are trying to train the T cells so that the trained T cells will not then or will recognize the peripheral tissue as self rather than non-self. So in this case, the male G expressing male tag cells, we were able to use our methodology and identify that it's very close to the MTAG2 cells, which express the ARE positive. And then we sort of confirmed that with IF staining. So I mentioned about other potential of using our methodology, site stereosec, to identify other niches. So in the thymus, there's also the B cells, and the functions of B cells in the thymus is still enigma. Not a lot is known about what they are doing, so we identify that they are clustered in the medulla regions, in especially the medulla 3 region, medulla 1 regions. So I mentioned about this HC structure, which is very unique to the human thymus, not found in the mouse thymus. So using our methodologies and our sort of pipelines, we were able to identify these HC regions to be aligning and co-localized with the medullary tree regions. And we sort of use maxima based on one of these markers that have been previously reported in the literature, S100A8, to sort of confirm our predictions. So again, we are doing a lot of niche analysis using our stereocytic samples, and I just want to maybe introduce a bit of our ongoing and future work. We really want to identify the unique niche and what RNA modifications is playing in some of these niche. And as I mentioned, we have quite a number of aging samples. We want to identify how thymus age. And from these informations, hopefully we could translate into how human embryonic stem cells or iPS cells could differentiate better to the thymic epithelial cells as well as CD4 positive T cells. So with that, I'll come to the end of my talk. I thank my collaborators, my lab members, and the funding agency. Thank you so much.
So talk is now open for discussion. Yeah, question? Yeah, there's my over there.
Basically, I have a suggestion. Why not just barcoding the M6A antibody with oligonucleotides, you know? Yeah.
Yeah. So were you asking how many? Or?
No, I think that we could just use the the total sig panel of the company of. Basically, we have you can label the M6A antibody with a specific oligonucleotides so that the poly-A tails. With a poly-A tail, so that the probes can also capture the signals from the antibody. Could that be a better method for capturing M6A modification signal together with other transcriptome information?
Right, it's a good suggestion. So I think you realize that our current strategies involve secondary antibodies, right? And therefore, you were suggesting tagging the primary antibodies and then trying to sequence. But then again, you need to make modifications. Unless the tag that you tag to the primary antibodies can somehow get into the cDNA. So with as in tagging the TM5 directly there, or
I think it's just with like the people doing the total sig panel. You can have an oligonucleotides with the poly-A tail targeted on the antibody.
Right. Yeah. So maybe we can explore more. I'm very interested to find out more.
Yes.
So let me introduce our third speaker of the session, Dr. Yogesh Huda from Child Health Research Foundation. We'll be talking about single-cell analysis of the nasal mucosa of children during respiratory infection. You have 10 minutes.
Thanks. I'll try to keep it to 10 minutes. Otherwise, we'll all miss our shuttle buses. So my name is Yogesh. I am a scientist at the Child Health Research Foundation, and I want to thank the organizers for giving me a chance to talk about our work. So I'm here representing my research foundation, which is based in Dhaka, Bangladesh. And our primary motivation is to look at infectious diseases in children and use that information to guide evidence-based policy decisions. And primarily, we do that by generating data on various kinds of infections that are common in children in Bangladesh. So these include systemic infections, neurological infections, respiratory infections, and all of this then is led to guiding policies for the various introduction of various vaccines. For the purposes of today's talk, I'll primarily be talking about respiratory infections. And the reason why respiratory infections are so important is because if you see all of the different like infections and other causes of death under 5 children in Bangladesh, about a quarter or like 24% of them is caused by pneumonia, which is a severe respiratory infections. And there are many pathogens and infectious agents that are involved in this, but the primary amongst them is this one particular virus called the respiratory syncytial virus. So it is something that infects children between the age of 2 months and 6 months. And it's very, very common, not just in Bangladesh, but many other LMICs. So this is a virus that we have been following for many years now. So starting from 2017, when we started doing RSV surveillance, we have been collecting samples from patients who have suspected RSV infections. And the counts are shown over in the gray. And in the red are basically the RSV positivity. So RSV follows this particular peak where, you have specific peaks where you have a large number of infections that occur. Like most other tropical countries, Bangladesh actually doesn't have a very specific RSV peak. So RSV can come like at various times in the year. Going next. So there are a lot of unanswered questions towards like RSV infections. So, we already know that RSV infections infect, particularly in LMICs, children younger than what we see in high-income countries. We see the infections are more severe. So about 97% of all mortality of RSV actually occurs in LMICs. And we see no seasonality. And again, similar to dengue, we don't have any way of triaging severe cases. And so when a child presents into the hospital, we have no way of knowing whether they'll require they'll require high support or will lead to death, or whether it'll actually be easily treated just by giving oxygen support. And so the question that we were interested in is to know, could single-cell genomics help over here? And so this is something that we could do as part of this CZI-funded project of the nasal mucosa, Global Nasal Mucosa Project. And this is led by, this is being done by 6 research groups in 5 countries and led by Jose Ordovas Montañez at Boston Children's Hospital. In this particular project, we are focusing on this one community. So the community is in a rural area called Mirzapur. This is located 60 kilometers north of the capital city, Dhaka. And over here, again, starting from 2007, our organization has been tracking a population of about 130,000 people. And this has a under 5 cohort, about 13,000 children. Now, in this particular population, a community health worker, which is a mother recruited from that community, visits every family once a week. And what they're looking for is they're looking for children that exhibit sign of sepsis, meningitis, or pneumonia. And these are then referred to the local hospital where we do all the testing and clinical microbiology laboratory work. And so using this or leveraging this ongoing large cohort study that we are doing, what we wanted to do is basically look whether we can actually study RSV infections using single-cell sequencing. And to really start that, this is Dr. Cheah. She's the co-PI of the project. And we started by community engagement. So here she is sitting with all the community health workers, the women in the community, to ask them questions about what would they be comfortable with, how would they want this study to be, and really co-designing the study with them. Next, we recruited a nurse and a porter from that same community. We got them to Dhaka and we trained them on sample collection as well as preservation. And then subsequently, we actually went back into the community to ensure that the families and the mothers especially are comfortable with all of the procedures that we are going to undertake. And this is really important because we were recruiting not just sick children, but healthy children as well as controls. And so this becomes even more important. In terms of training for single-cell sequencing, again, because of this collaborative effort, I got an opportunity to go to Boston and train in various different sequence capture methods. And finally, based on the fact that it doesn't require any machine instrument or any other effort, we focused on the honeycomb system. And then subsequently, I was able to bring that to Bangladesh, train my team, as well as people from Jose's group could come in, and we could really established the single cell capture and sequencing methods in Bangladesh in our lab. Once we had this sort of system set up, we started tracking for the RSV season. So this was something that we really waited, and in September 2023 is when we started seeing a RSV peak. And so this is kind of when we started collecting samples. So for the sample, we would send the porter and the nurse into the community. They'll collect the sample from the child. And then from the same community, an age-matched, sex-matched healthy child is selected. And then a sample is also collected from that child as well. Now, apart from that, like we actually had to figure out how to get the dry ice, how to get Mr. Frosty into the dry ice, collect samples in the community, and then these samples were shipped to the local hospital, which had a -80 freezer. They were all stored there. And once all the sample collection was done, all of these were shipped to Dhaka on dry ice, where we really used them for single cell capture and subsequent downstream analysis. So far, we've collected about 170 swabs. So that includes 85 children with potential RSV infections, 85 healthy children. And we've had 100% consent rate for sick children and about more than 80% for healthy children. Out of this, we have now sequenced 17 swabs, giving us 23,000 cells from these samples. And these include 6 healthy, 5 sick but not RSV positive, and 6 RSV positive samples. We also contextualize these with 11,000 cells generated from 4 healthy children in Boston, and this was also done on the Honeycomb platform. And so this is the data, all the data that I'm talking about here. So this is just to show that we get very similar cell populations in Boston and in Bangladesh. We can annotate, so we can identify the different cell-specific clusters, we can annotate them, and all of them like are cells that you would normally see in the nasopharynx. So we know that we are being able to collect the samples effectively, bring them to our lab, and actually conduct single-cell sequencing, which is really, really amazing. And then, once you have this, obviously, the next question is, what can we learn about like RSV biology? So over here, I'm showing you on the UMAP all the cells that came from healthy individuals, sick individuals, as well as RSV positive. And over here, I'm showing you all the cells that have an RSV RNA expression. And you can see that, you see as seen by, like in Boston, as seen in Jose's group, we also see that, we have a large amount of RSV in plasmatoid dendritic cells, as well as a subset of secretory cells that also express a lot of immune markers. So this is something that we are now pursuing further to see what are the various markers or what are the other genes that are involved in this. But other thing that we were particularly interested in was to see whether we have five cases where we have a sick child, but we don't know what's actually causing it. So could we use all of the single cell sequencing data to predict what are the kind of viruses that exist in the community among sick children? And so for that, we used a metagenomics tool, Kraken, and a RefSeq viral database. And we made basically an average of all the healthy samples. And then using that, we try to predict what are the viruses that are potentially enriched in sick children. Not surprisingly, 4 of the 6 samples had RSV. I mean, we know these from PCR positive cases. But we also saw this one particular Orizo-pox virus. This was also present in some healthy children and also has been seen in Boston. And additionally, we also see human parainfluenza virus, which is something that we did not know about. But now we can easily design PCR assays and really go back and see how common or how prevalent this particular virus is in this community. And so, tools like this, methods like this really allow us to sort of develop and study the kind of respiratory infections that exist in our communities. And obviously, like, this is an ongoing project, so we are still sequencing more, and we are going to do a comparison of, the data that we generate in our lab versus the data that is generated in Boston and all the other places where this study is being done. We are also we've collected a lot of clinical as well as like environmental factors. So it'll be very exciting to see how that compares with the data that we get. So far in this one, this is something that, this is a study that was done in the community. So a lot of these children had symptomatic infections, but they were not severe. So in the next phase of the study, we actually want to take hospital cases of RSV to really see if these severe cases have any specific cell differences, and could we use that to like predict, like kind of like identify prognostic markers for this. And what we're really hoping to do with all of this is to better understand the pathophysiology of this particular viral infection, and hopefully other respiratory infections that affect Bangladesh, but also other places in the world. And with that, I'd like to thank basically all the mothers, all the community that really participated in this project. These are our sample collection team, which really went in and did all of the collection, and also the lab team who did all of the sequencing, all of the analysis. And these are all nestled between, we are a large organization that really makes all of this work possible. And also, so I want to thank all the people who made, really supported this work, including the funders, Bill and Melinda Gates Foundation and the WHO, who really help us set up this cohort and this community surveillance, and Chan Zuckerberg Initiative for the funding for this specific project. Thank you so much.
Thank you so much, Yogesh. We have time for only one very short question. I'm on my toes now. Okay. If there's no question, thanks so much. So I'll close the session and hand over to Angela. But please be reminded, don't post any of the unpublished data for all the talks on the social media.
Okay, I will make this quick so we can all make the shuttle back to the hotel. First of all, thank you all so much for being here and staying until the end. It's been a great meeting. I've really enjoyed it, and I hope you've all enjoyed it as well. Just a few numbers for you to appreciate. I think some people were asking about this. We have attendees from 31 countries, and including the virtual attendees, we had over 400 people watch this meeting, which is far, far, far exceeded my expectations. So thank you all again so much for your support. Thanks to all the poster presenters. Yeah, thank you. Thanks to all the poster presenters. Thank you to all the speakers and the chairs, and you gave wonderful talks. And of course, all your participation in the breakout sessions. In case you're interested about the makeup of attendees from the different countries, China was really representing this time. But we also had a strong cohort from Korea that you can see there. Key takeaways. I think in the interest of time, we'll go quickly on this, but I think during the breakout sessions, many of the points that were raised by Aviv and Sarah in the introduction were echoed, that there are plenty of opportunities to gather funding for collaborative research on diseases that utilize tools like single-cell and spatial omics, and I think hopefully there were collaborations formed here to do that. There is a wide availability of FFPE samples, so HCA researchers can really leverage these new spatial technologies that we've been hearing about. And a lot of new participants this time at this meeting, which is also fantastic. I think we also heard a lot about diversity, which we obviously as a group care a lot about, and we're continuing to push that. This is something that was brought up last year and continues to be a theme, I think, and will be for the following meetings as well. In particular, we would love to engage the researchers from mainland China. I think some of them came to talk to me during the breaks, expressing an interest in contributing and working together. So I think we can follow up on that. And yeah, please join a BioNetwork and talk to any of us if you want to get more involved. We do want to hear from you. There's a 5-minute survey that you can take on your own time. You can scan the QR code now and do the survey later. I'll give you a second to do that. All right. The recordings are also available for both the 2 days. So if you're registered, you have access to the online recordings. at this link. I think you can find this on our website as well. Special thanks. Okay, I don't want to rush this part too much. We want to again thank our sponsors, 10X being the biggest one. Thank you to MGI, to CZI, Scale Biosciences, our other sponsors and exhibitors who you saw out there. Thanks to their support, we can have this nice venue to hold this conference. Again, CZI, the Klarman Family Foundation, Wellcome Trust, and also HKUST, actually they've given me enough funding to be matched to what 10X gave us. So I also want to just give a shout out to them. And also the Center for Translational Stem Cell Biology, which gave us support so that we could be at this venue. Huge thanks to the committee over the year. They're the ones who selected the speakers and invited the speakers to come. Al, Lan, Partha, Pompan, you saw Pompan, Shyam, and Jay, obviously, Yuki, Woonyang, who couldn't be here, Senjudi, Shigong, who I think also had to leave early, and Jay. Thank you all so much. And then special shout out to Mu, who not only brought in money, but was helping me along all the way. And Jiguang is our third local coordinator. Speakers, session chairs, panelists, I think reading the names will take too long, so I'll just pause here for a moment so you can appreciate their contributions. Thank you again. And all the poster presenters, thank you, great posters. And also our volunteers who come from HKUST and Hong Kong U. They're the mic runners and scribes for the different events. So thanks to the students back there. Okay, event management group, thank you guys. AV, the venue, Science Park, Queenie. And special thanks to Yoshi and Esther, who I think without them I would not live. Thank you so much. And then after this, I'm passing it to John.
I want to thank everybody that Angela just mentioned. I also want to thank Angela because she cannot thank herself. So thank you very much for your leadership in putting this meeting together. Just really quickly, some upcoming events, the biggest one being our next annual general meeting. This is sort of the big annual meeting for all of HCA. It will be in Singapore next June, and so please attend. Registration is now open, and you can register with this QR code or just go to our website. This is a very nice picture to inspire you to come. Xiao and Jay in Milan said something to the effect of, come for the food, stay for the science, or maybe it was come for the science, stay for the food. I don't know, but either way, please come. Anyone can contribute. You can use this QR code to join HCA. Please do join HCA if you are not a member. Anybody can join. There's no cost. And once you do join, then you'll receive updates on HCA activities. And you can also then join one of the biological networks. In fact, you can join more than one. So join one or more biological networks. Connect via our Slack channel. Again, you can find all of this on our website. Until next time, thank you all very much, and please have a great day.